हेलो। हेलो। बता रहा हूँ अभी मैं, क्लास 10 बजे से बता रहा हूँ, 10:05 पे। ठीक ठीक।
ये लेक्चर ख़त्म होने के बाद कैसे ये करना है?
कुछ नहीं, वो मैं अपने आप कर देता हूँ आज।
नहीं मतलब क्योंकि इसमें एस्केप वगैरह कुछ होता है ऑप्शन या नहीं?
नहीं, आपका जैसे ही ख़त्म होता है ना तो बस वो मैं तो टाइम जैसे मैं फ़ोन पे देखता रहता हूँ, जैसे 12 12:15 बजे जैसे भी ख़त्म होता है ना मैं आके फिर इसको बंद कर देता हूँ।
नहीं नहीं, अब ये देख, ये ना मेरा ओपन हो गया ना अब।
हाँ जी।
तो अब ये मुझे बंद करना है तो कैसे करना है?
यहाँ से ये इधर से ये आया ना,
ये यहाँ से?
ये ऊपर वाला ये वाला।
ओके।
अभी है 7 मिनट है अभी तो, क्योंकि मैंने अभी कैमरा वॉइस ऑफ किया हुआ है।
कैमरा भी ऑन। अच्छा। कहाँ गया वो? ये कैमरा भी ऑफ ही है। स्क्रीन शेयर्ड है ना अभी तो?
वो मैंने किया हुआ है।
शेयर किया हुआ है। हाँ पता, हाँ झालं काय सानूचं? अच्छा हाँ ठीक आहे, ते मॅडम मग हे करते. ठीक आहे, रडली नाही ना सानू? सानू रडली नाही ना? बरं. ठीक आहे चालतं, ठेवू मग? द्या बरं तिला, सानूला द्या. हाँ हाँ बेबी, व्यवस्थित लक्ष दे बाळा हाँ क्लासमध्ये, ठीक आहे? ठीक आहे बाबू? हाँ ठीक आहे, खाल्ला ना तू? बोल पटकन. हाँ ठीक आहे, मी लेक्चर घ्यायला लागले, संध्याकाळी आपण भेटूया, ठीक आहे? ओके चल बाय.
आपका 10 से 12 रेगुलर या 10 से 11?
10 से 11 है मेरा.
और 11 से कौन आएगा? 11 लीटर
जरैल सर का है. जरैल सर का.
जी सर.
तो इनका अटेंडेंस कैसे होता है?
टेलीमेडिसिन से होता है मैम, बताइए.
अच्छा. तो इसमें बाकी के कोई भी जो है, अगर ये यूट्यूब है, यूट्यूब पे आता है तो कोई भी ज्वाइन कर सकता है इसे? ये कैसे होता है?
अगर जो उनको लिंक पता होगा तो
लेकिन लिंक पता हो तो ज्वाइन कर सकते हैं?
हाँ.
इसका मतलब कोई भी ज्वाइन कर सकता है मतलब?
नहीं, उसको लिंक पता होना चाहिए. यूट्यूब का अलग लिंक है इसमें और गूगल मैट का अलग.
हाँ लेकिन ये बच्चे तो दे सकते हैं ना किसी को भी.
हाँ यूट्यूब का भी लिंक दिया हुआ है. वैसे बच्चे है ना, मैंने कई बार देखा है इसको, इस पे ही वो देखते हैं, गो टू मीटिंग पे.
अच्छा अच्छा, उन्हें फिर क्वेश्चन पूछना होता है ना?
हाँ जी. इसको मैं कर देता हूँ आपका, इस पे डालना था ये. अभी करते हुए इसको ऑन कर दें।
ठीक
ठीक आप देख लें।
Good morning everyone. Can you hear me?
Yes, ma'am. Yes, ma'am.
Okay. How are you all? You all are fine?
Yes, ma'am. Fine, ma'am.
Okay. So we will wait for 1-2 minutes and then we will start. So your classes started for anatomy and biochemistry also, है ना?
Yes, ma'am.
Okay. So let's start the lecture. So good morning everyone. Today we will see what is erythropoiesis and how this erythropoiesis is regulated. So till now we have seen what is the blood. So what are the blood? What are the elements which form the blood? So there are three important cellular elements like RBC, WBC and platelets. Okay. So these cellular elements play the main part or around 43 to 45% of the blood. Out of that, RBCs form the most of the chunk of cellular element is formed by the RBCs. Okay, so their count is around 4.5 to 5 millions per cubic millimeter of blood. So can you guess how many RBCs are formed each second? Can we guess?
Ma'am, we cannot see any PPT.
I am not showing any PPT, but you can see the first slide, erythropoiesis and its regulation. Is it visible? Can you see
No,
it?
ma'am, nothing is visible.
Nothing is visible.
No, ma'am. No, ma'am. That's
Can you see now? Can you see anything?
yes, ma'am. Yes, ma'am.
How to minimize it? So that go-to meetings, matlab that logo is also visible. What is visible to you?
Just the PPT.
Just the PPT, na? Okay. ठीक है. So ठीक है. So can you guess how many RBCs are formed each second? Anyone can guess? So around 2.4 million RBCs are formed each second. Okay, so can you imagine that 2.4 million RBCs are developed by our bone marrow each second, each day? That means that is a huge amount. Okay, so the number is so much, and so as the number is more, we also need the tight control over this process. So it is the well organized event, that is erythropoiesis. That is how the production of RBCs will take place and how it is regulated. So let us see today what is the function of bone marrow, what is the function of erythropoietin in this erythropoiesis. So the objectives of today's lecture are: at the end of the lecture, you should be able to describe the maturation sequence in the development of erythroblast to mature erythrocytes, and you should be able to describe the site of production for erythropoietin and stimulus for its synthesis, because erythropoietin is the main hormone which is involved in erythropoiesis, which is very important or critical for erythropoiesis. So you should know where it is produced and how it acts over the progenitor cells, which leads to erythropoiesis. So in the last lecture we have seen where the hematopoiesis takes place. So erythropoiesis is a part of hematopoiesis. So the site will remain same. That means during prenatal period it varies. The site will vary from the age of gestation, and after birth the erythropoiesis takes place mostly at the bone marrow. And in the bone marrow also, initially the long bones, the bone marrow of long bones is involved in hematopoiesis, but as age advances, the cellularity of bone marrow will go on decreasing and it will get replaced by the adipose tissue. So at the age of 30 years, most of the hematopoiesis takes place only in the membranous bones, which are the sternum, ribs, okay, and then pelvis, vertebra. So only at these places the hematopoiesis will continue after the age of 30 years, and cellularity of this bone marrow also goes on decreasing as age advances. So there are this is what we have discussed in the last lecture, that is during fetal life there are 3 stages. So mesoblastic stage, which is for 2 months, then hepatic stage, that means the stem cells move to the liver and there hematopoiesis takes place. So spleen also takes part in this hematopoiesis during mid gestation, and then myeloid stage during last month, that is red bone marrow and liver. Now after in the newborns only red bone marrow is involved in hematopoiesis, and in children up to 6 years red marrow of all bones is involved in hematopoiesis. Then from 6 to 20 years again the changes will take place, and red bone marrow of long bones and all flat bones are involved, and in adults mostly the membranous bones are involved in hematopoiesis. If there is any disorder, if there is hematolysis, if there is any condition which is affecting bone marrow, then hematopoiesis will shift from bone marrow to liver and spleen. So that is called as extramedullary hematopoiesis or erythropoiesis. So what is erythropoiesis? So erythropoiesis is the process of red cell formation in the bone marrow, and it involves variety and number of cells at different stages of maturation. So in the last lecture we have seen that it starts with pluripotent hematopoietic stem cells, then it will go to the cells will differentiate and multiply and they form the progenitor cells, and then again they differentiate and multiply and ultimately they lead to formation of the mature cell. So the erythropoiesis involves a variety of and number of cells at different stages of maturation, that is from stem cell to RBC. So for descriptive purposes, erythropoiesis can be divided into 3 stages. First stage is commitment, that means the pluripotent cell differentiate and divide into committed cell. Okay, that means that committed cell that can develop only into RBC. So that is the meaning of committed cell. It cannot revert back to the pluripotent hematopoietic stem cell, but when it differentiates and when it divides, then ultimately it leads to a mature form of cell, and in this case it will lead to formation of RBC. So this is the first step is commitment. Then early phases of differentiation, and that is that stage is erythropoietin independent. Okay, so even though erythropoietin is required for erythropoiesis, the early phases of erythropoiesis are erythropoietin independent. They have a few receptors, but the survival is not dependent upon erythropoietin. But the late phases they depend upon erythropoietin completely. So their survival also depends upon the presence or absence of erythropoietin. So we will discuss it in detail. So let us start with the erythropoiesis. So the red so it is important to have tight regulation over this erythropoiesis, because the rate of formation is very high. Okay, 2.4 million cells per second. So this rate is very high. So if it goes uncontrolled, so what will happen if it more and more RBCs are formed and the destruction is not that much, but the synthesis is so much. So the whole of our blood, it will have only RBCs, and it will affect the blood flow. It will affect the viscosity of blood. It will increase the resistance to the blood flow, and all the system will get filled with the RBCs. So it is very important to have the tight regulation of this erythropoiesis. Okay. So there are control mechanisms which can regulate this erythropoiesis, and erythropoietin is the major humoral regulator of RBC production. We will see what is the importance of erythropoietin. So it is a tightly regulated process. So in brief, this is the process of hematopoiesis. So these are on left side we have pluripotent hematopoietic stem cells which differentiate into colony forming unit spleen, which again, so this colony forming unit spleen still has a stem cell properties, so it can divide, it can give to multiple lineages. So one of the lineages colony forming unit blast, it is also called as burst forming unit erythrocyte. So there are 2-3 names to the one cell, so you have to remember it. So colony forming unit blast is also burst forming unit erythrocyte, which again differentiate into colony forming unit erythrocyte. So these two cells are called as progenitors. Okay, so these are the progenitors, and then they differentiate into precursors. Now what precursors we will see in detail. So just remember at this stage that colony forming unit blast or burst forming unit erythrocyte and colony forming unit erythrocyte. So these two are the progenitors for erythropoiesis. So what is erythron? So erythropoiesis involves, we have seen that large number of different stages of cells at different stages of maturation, from stem cell progeny committed to erythroid differentiation to mature circulating red cell. So that is colony forming unit blast cells and colony forming unit erythrocyte. From that progenitors to red blood cells, mature red blood cells which are circulating in the circulation. So that whole mass is considered as a family, and that whole mass is given a name, and that name is erythron. Okay. So when we talk about erythron, so it includes all the cells from progenitors to the mature circulating RBCs. So that is one family, and that is the called as erythron. Now let us see the characteristic of each cell. So we will start with the progenitors. So there are two progenitors in erythropoiesis, that is colony forming unit erythroid and burst forming unit erythroid. Okay, now so what is the importance of these two progenitors? So what is colony forming erythroid? What it does? So it depends upon the presence of erythropoietin. The first thing is, so it is very sensitive for the presence or absence of erythropoietin, because its survival depends upon erythropoietin. It has large number of erythropoietin receptors over its surface. So under the influence of erythropoietin, it will give rise to around 8 to 32 well hemoglobinized cells. So how it is understood means how we know that number, because this when when these cells multiply in the culture media under the influence of erythropoietin, then they will give rise to 8 to 32 well hemoglobinized cells. Okay. So what is the characteristic of this colony forming erythroid cell? So they appear as immature cells when we stain them in the bone marrow slides, and they have fine nuclear chromatin because they multiply actively. They have well defined nucleoli as there is cell division takes place. Then higher nuclear to cytoplasmic ratio, so size of the nucleus is very high, and they have basophilic cytoplasm with pseudopods. So basophilic cytoplasm indicates that there is the number of ribosomes which are involved in protein synthesis is very high, and so they are basophilic. Okay. So what is the characteristic of this cell? So it is the most erythropoietin sensitive cell, and it has highest number of erythropoietin receptors on its surface, and it depends upon erythropoietin for the survival. So erythropoietin, which is very important hormone required for erythropoiesis, it will act on this cell. So this is a progenitor cell, so it will act on this cell, and it will initiate the differentiation of the cell. So you must be knowing what is the difference between multiplication or division of the cell, and what is the difference between differentiation. Okay. So in differentiation, so in multiplication, both the daughter cells will be same as mother cell. Okay. But in differentiation, some changes will take place in the daughter cell when the cells multiply and divide, so that the cells which are resulting after the division, they are different from the mother cell. Okay. So they are different from the original cell. So this erythropoietin will start the differentiation in this progenitor cell, and it will also, and when the differentiation starts, it will also start the synthesis of hemoglobin. So hemoglobin synthesis will start from these progenital cells. Now the next progenitor cell is the burst forming unit. Ideally the burst forming unit erythroid, it develops from pluripotent stem cell. Okay. So it will come first, but for explanation purpose we have taken it as the second one. So it is less mature than colony forming unit erythroid, because it develops immediately from the pluripotent from the stem cell, and it closely resembles the multipotent hematopoietic stem cell. So structurally it resembles, but when we see the expression and all expression of these cells, so the expression of different proteins, they are different from the hematopoietic stem cells. So it is smaller than colony forming unit erythrocyte, and it means it is immature blast cell which has basophilic cytoplasm, occasional pseudopods, and clear nuclear chromatin. Now, so what is the characteristic of this cell? So it requires erythropoietin, okay, but its survival depends upon another factor that is called as interleukin 3. Okay, so there are growth inducers and differentiation inducers. Growth inducers will cause multiplication of the cells, and differentiation inducers called differentiation of the cell. So erythropoietin induces the differentiation, and this interleukin 3 acts on the progenitor cells. Okay, so the burst forming unit, it depends upon its depends upon interleukin 3 for its survival, and it also requires other factors like interleukin 6. So all these are growth factors like interleukin 6, colony forming factor, granulocyte, macrophages. So all these factors are required, and in the presence of all these factors, it will give rise to around 500 to more than 30 to 40,000 well hemoglobinized erythroblast. So it is called as burst forming. That means the number of cells which can be developed from this cell is very high. So it is called as burst. Okay. So colony forming unit erythroid, it will lead to formation of limited number of cells. So only 8 to 32 cells can be formed from the colony forming unit erythroid, but burst forming cell, they can lead to form form around 500 to 30,000 to 40,000 erythroblasts. Okay, and this is a initial stage of erythroid differentiation, and it is independent of erythropoietin. So in patients having chronic renal failure, so in these patients also these cells are present. That means they don't depend upon the erythropoietin for their survival. They need small amount of erythropoietin, but their survival depends upon interleukin 3. So what is the summary of these progenitor cells? So during erythroid development, early progenitor cells have the very high proliferative potential, and they depends upon interleukin 3, and their response, they need erythropoietin, but mostly they are depend upon interleukin 3, and they differentiate through various stages into later progenitor cells which are interleukin 3 independent, and they are completely dependent upon erythropoietin. So erythropoietin acts on the later stages of progenitor cells, and the initial stages are independent of erythropoietin. So now once we cover the progenitor cells, now we have the erythroid precursors. So when we discuss about erythroid precursors, so again the nomenclature is, we have 2 to 3 types of nomenclatures. So we can say the cell as erythroblast. So what is erythroblast? So erythroblast is referred to all forms of mature red cells, all forms of not mature, all forms of cells, red blood cells which have the nucleus. Okay, if the RBC has the nucleus, then we tell that RBC has the nomenclature for that RBC is erythroblast. The mature RBC will not have the nucleus. So these are all immature cells which have the nucleus. So normally if that cell is normal, then it is called as normoblast, and if it is abnormal, mostly it is large in size, so it is called as megaloblast. So the least mature precursor or the precursors will start from the pronormoblast or proerythroblast. So you have to remember all these stages in sequence. Okay, so if you get one question on what are the stages of erythropoiesis, then you have to write all the stages in sequence. Okay, that is very important. you cannot write first reticulocyte and then proerythroblast. So you have to start with proerythroblast, that is the least mature, okay, or most immature precursor of the erythrocyte or RBC. So the stages of erythropoiesis will start with proerythroblast, then early normoblast or also called as basophilic erythroblast, then intermediate normoblast or polychromatophilic erythroblast, then late normoblast or orthochromic erythroblast, then reticulocyte, and then mature RBC. Now let us see about the so this is a picture of stages of erythropoiesis which is given in Guyton. So you should know what is the trend during the morphological development of these RBCs. So as we proceed from proerythroblast towards RBC, the size of the cell will go on decreasing, the size of the nucleus also goes on decreasing, and at one stage the nucleus will be removed or expelled from the cell, and the reticulocyte and mature RBC will not have the nucleus. Now the nuclear chromatin will first, initially it is clear, and then it will start the condensation of this nuclear chromatin will start, and again at the orthochromic stage it will become highly condensed and it will be expelled out, and then reticulocyte and RBCs don't have the nucleus. Then the characteristic of cytoplasm will also go and change. So initially it is basophilic, as it has high content of ribosomes, so because the protein so it is actively synthesizing the protein, okay, so it has high amount of ribosomes and it gives its basophilic appearance. As so initial from the precursor stage also the hemoglobin synthesis will start, and at polychromatic stage the hemoglobin level increases so much that it will change the staining characteristics of the cytoplasm. So it will start appearing as pink, and then means cytoplasm, the color of the cell will change as the content of the cytoplasm changes. So we will discuss in detail. So this is pro-normoblast, and that is the most immature cell. So the size of this cell is 12 to 20 micrometer and round, or it is slightly oval in it is round in shape. The nucleus is also round or slightly oval, and it has nucleoli as it undergoes cell division, so it has nucleoli and chromatin is fine, and the nuclear to cytoplasmic ratio is very high, it is 8:1, and cytoplasm is dark blue, okay, and it may have the prominent Golgi apparatus because it actively synthesizes the proteins. So in the bone marrow it is around 1% of the cells are formed by this pronormoblast, and we don't have pronormoblast in the peripheral blood. So this is the most immature cell in this erythropoiesis. Now next cell is basophilic normoblast. So I have already told you that is it is called as basophilic because its cytoplasm is highly basophilic. You can see here it is dark blue cytoplasm, as it has large very high content of ribosomes. Then the nucleoli are decrease in number, 0 to 1 nucleoli are present, and chromatin is slightly condensed. So here you can see in the nucleus, you can see the areas where the chromatin starts, the condensation of chromatin has started, okay. So the number of these cells in bone marrow is 1 to 4%, and again we don't observe any basophilic normoblast in the peripheral blood. So the next cell is polychromatophilic or polychromatic normoblast. Now it is called, so can you appreciate the difference between the cytoplasm of basophilic normoblast and polychromatic normoblast? Okay, so what is the difference? What is the difference between basophilic and polychromatic normoblast? Okay, so the staining characteristics change because now polychromatic normoblasts, they have sufficient amount of hemoglobin, because hemoglobin synthesis continues, okay, from progenitor cells. So it has accumulated so much hemoglobin that hemoglobin will give its own staining characteristics. So hemoglobin is acidophilic in nature. So when we stain with Romanovsky stains, so it will take that acidophilic stain and it gives pinkish color. Okay, so it is called as polychromatic normoblast. So it is the cell from which visually we can say that hemoglobin synthesis, hemoglobin is present in the this bone marrow cell. Okay, then what happens in the nucleus? So during these stages, function of the nucleus goes on decreasing. Okay, and because the hemoglobin has the effect on the nucleus. Okay, so it decreases the function of nucleus, the chromatin will start condensing, and it is in this polychromatic phase, it is quite condensed, chromatin is seen here. So the function of nucleus will go on decreasing as hemoglobin starts accumulating in the cell. Okay, so cytoplasm is grey blue as a result of hemoglobinization, and nuclear to cytoplasmic ratio also decreases. So size of the nucleus also decreases, and it is the most common cell, or it is a in this all the progenitor in all these cells, it is the highest number of cells which are observed in the bone marrow. So 10 to 20% bone marrow cells are polychromatic normoblast. Again, we don't observe any polychromatic normoblast in the peripheral blood. Now the next stage is orthochromic normoblast. So here the nucleus nuclear chromatin is fully condensed. So because it will, and because as the nuclear chromatin is fully condensed, it cannot divide, or it cannot leads to replication of DNA. Okay, so at this stage the multiplication will stop, and the nucleus, the cytoplasm, you can see it is more pink because hemoglobin is accumulated inside the cytoplasm, and at this stage the nucleus will be extruded or expelled out from the cell. Okay, so there is another process for expulsion of the cell, it is not required for you, but from orthochromic normoblast, the nucleus will be removed from the cell, and once the nucleus is removed, the cell cannot divide or it cannot produce proteins. Okay, so in bone marrow we observe 5 to 10% cells as orthochromic normoblast. So the next one is the reticulocyte. So here you can see that the size of reticulocyte is higher or larger than the mature RBC. So on right side we have the mature RBCs. So the size is higher than the RBC. It don't have the nucleus, but some cell organelles are still present, like ribosome, mitochondria, Golgi apparatus, they are present in the cytoplasm. So once the nucleus is removed from the cell, these reticulocytes will remain in the bone marrow for few days. Okay, where again maturation of these cells takes place, the cell size will decrease, and also the remnants of the cell organelles, they will be removed, and then the cell will be converted into mature RBC. Now the number of these cells is very less because the lifespan of this cell is very less. So within 1 to 2 days, these reticulocytes, once they enter the peripheral circulation, they will get mature into mature RBCs. Okay, so their number is very less. So in the peripheral blood we observe reticulocyte. So you will perform the practical in which you will in your own peripheral blood, you will do the staining of the your blood slide, and you can observe your reticulocytes in the peripheral blood, but their number is very less. It is only 0.5 to 2%, because within 1 to 2 days they get mature into RBCs. Now why they are called as reticulocytes? Because once they are formed, they still have the cell organelles like ribosomes and Golgi apparatus, and if we stain these cells with suprametal stain, these ribosomes and they get precipitated and they form a reticulum like structure. Okay, you can observe it under the microscope. So as they form the reticulum like structure, it is called as reticulocyte. So reticulocyte and mature RBCs, they don't have the nucleus, even though they have the enzymes which are required for the normal functioning of the cell. So even though RBCs don't have the nucleus, the RBCs can survive up to 120 days. Okay, that is very important part. So it has the machinery which is required, or the enzymes which are required for maintenance of the survival of this cell for 120 days. So these are all the steps in erythropoiesis. You should know by name, and with the characteristic of each cell you should be able to write the question on this erythropoiesis. Okay, this is a very important part, and you should know what are the different characteristics of each cell, and how they proceed from stem cells to the mature erythrocyte. So when the erythropoiesis is stimulated, so each pro-erythroblast will give rise to around 8 to 12 mature red blood cells. Okay, and most of the cell division takes place only up to polychromatic erythroblast. So up to polychromatic normoblast, the cell division can take place, and orthochromatic erythroblasts cannot divide because they cannot synthesize DNA or cannot replicate DNA, and so cell division is not possible after orthochromic normoblast. Okay, any question? Do you have any question till this part? So it is very simple. Once you read, you will understand it. You have to remember the basic concept that as the cells go on dividing, there will be differentiation and there will be cell division. So the size of the cell goes on decreasing, the size of the nucleus goes on decreasing, the nuclear chromatin will go on condensing, and the staining properties of the cytoplasm will change, and ultimately the nucleus will be removed from the cell body, and there's only the RBC is nothing but a bag like structure which has the hemoglobin in it. So it carries hemoglobin. So the main function of RBC is to carry hemoglobin. Okay, so it don't have the nucleus, and so hemoglobin will perform all the other functions. So let's proceed further. Okay, so now as I have told you, as the differentiation of progenitor cells start, simultaneously as they differentiate into different progenitors, the hemoglobin synthesis also starts. So there is expression of the genes which are involved in hemoglobin synthesis. So hemoglobin forms the 90% of the dry weight of the RBC, and it is intimately related to the erythropoiesis, and also so differentiation of colony-forming erythroid into erythroid precursors involves activation of genes which are related to hemoglobin synthesis, and it has 3 complex metabolic pathways which we will see tomorrow in next lecture. And so hemoglobin, what is the role of hemoglobin? Hemoglobin also it has inhibitory role on the nucleus. Okay, so once the hemoglobin starts accumulating inside the cell, it will move to the nucleus and it will inhibit the nuclear function also. Okay, so if hemoglobin level is less, suppose the patient has the iron deficiency anemia. So in iron deficiency anemia, the amount of hemoglobin formed is less. So what will happen? So the cell will go on multiplying and dividing. Okay, so as the cell goes on dividing, the size of the cell will go on decreasing. So what we observe in iron deficiency anemia, the cell size is less, which is called as microcytes. Okay, if the cell size is less, it is microcyte. So microcytic and hypochromic anemia is present in iron deficiency, because as the iron is deficient, the hemoglobin level which is formed is also less, and the cell will go on dividing, because hemoglobin, the hemoglobin also inhibits the nucleus. If the large amount of hemoglobin is accumulated inside the cytoplasm, the nucleus will be removed from the cell. Okay, so if the hemoglobin level is less, the multiplication continues and the cell size will go on decreasing, which leads to microcytosis. We will discuss again during the lecture on anemia. Okay, so how this erythropoiesis is regulated? So the most important hormone which is regulating erythropoiesis is erythropoietin, which is secreted by the kidney. Now another hormones, they also play important role, like androgens or testosterone. They will stimulate erythropoietin production. They potentiate or increase the population of stem cells on which erythropoietin can act, and they stimulate the erythropoiesis. And on the other hand, estrogens have the inhibitory role. So they suppress erythropoietin production and they also inhibit stem cells. So because of this, we observe the gender differences, that is in males and females the RBC count or hemoglobin level is different. Okay, so males have higher number of RBCs and hemoglobin level is also very high as compared to females. So because this androgen has stimulatory effect and estrogens have inhibitory effect on erythropoiesis. Now this we also need other hormones like thyroxine, cortisol and growth hormone, which have stimulatory or effect on erythropoiesis. So during your endocrinology, you will see what is the role of other hormones on erythropoiesis. Now products of now there are also important factors are dietary factors. So dietary factors have the important role in erythropoiesis, because the large number of cells are formed every day. So if we have deficiency of these factors like iron, protein, vitamin B12, folic acid, if we have the deficiency of these factors, then they will critically affect the erythropoiesis. So we will see in detail during the lecture on anemia. So all these factors will regulate the erythropoiesis. So what is the so the main important factor out of all these is erythropoietin. So what is the stimulus for secretion of erythropoietin? That is tissue oxygenation. So if there is decrease in the tissue oxygenation, so decrease in tissue oxygenation may be because of decreased blood volume, or if the patient has anemia, that is low hemoglobin level, or if the blood flow is poor to the kidney, or if there is any pulmonary disease or cardiac disease which is affecting oxygenation of blood. Okay, so all these factors will decrease the oxygen level of the kidney or other tissues, and it will lead to production of erythropoietin. Okay, so kidneys will form the erythropoietin, and this erythropoietin then will act on the proerythroblast, means the colony forming unit erythroblast, and that will lead to increased differentiation of these cells and multiplication of these cells into proerythroblast and again to RBCs. So then it will increase the erythron size of the erythron, and then it will increase the tissue oxygenation. So it is a negative feedback mechanism which will be which is acting here, and it will then again restore the tissue oxygenation level. So tissue oxygenation level is the main stimulus for the release of erythropoietin from the kidney. So so again the same thing, that is erythropoietin induces erythroid progenitor cells differentiation into pronormoblast, and it brings about the expansion of erythroid mass and RBC production. So ultimately increases the size of erythron, that is from progenitor cell to the RBC. It will increase each the number of each cell and ultimately increases the size of erythron. So from where this erythropoietin is secreted? So it is secreted from the peritubular fibroblast like cells which are present in the kidney. So these cells will secrete the erythropoietin when there is hypoxia. So which are the factors which are necessary for erythropoietin? Okay, so necessary means without these the erythropoietin will the erythropoiesis will be affected. So there are some general factors and then some specific factors. So general factors are erythropoietin, thyroxine, hematopoietic growth factors like interleukin 3 and then stem cell factor, granulocyte, monocyte, choline-stimulating. So all these growth factors are also required, and vitamins B, C, D, E. Okay. So these factors are required for the hematopoiesis. Now maturation factors. So maturation of RBCs depends upon vitamin B12, intrinsic factors and folic. So for the time being just remember these. So like we need the maturation of RBCs, and there are few factors which are required for the maturation of RBCs. So we will discuss these factors in detail during the lecture on anemia. Okay. So these are the maturation factors which are required for maturation of RBCs. Now factors necessary for hemoglobin synthesis. So these are the proteins, because hemoglobin has globin chains which are nothing but polypeptide chains. So proteins are required for synthesis of hemoglobin. Iron is required. You all know that heme part which is present in the hemoglobin, it has iron in it. Then we need copper, cobalt. Vitamin C is required for absorption of iron from the intestine, and we also need riboflavin, nicotinic acid and pyridoxine. So all these factors are very essential for erythropoiesis. Suppose we have iron deficiency or protein energy deficiency, malnutrition, then we will develop the anemia. And also if we have vitamin B12 or folic acid deficiency, then also will lead to anemia. And also in patients who have chronic renal condition like chronic renal failure, then we have the deficiency of erythropoietin, and deficiency of erythropoietin leads to decreased synthesis of RBCs and the patient develops anemia. So it is very important to give erythropoietin to the patients who have chronic renal disease. Okay. Any questions? Do you have any difficulty in understanding some part or any question related to this topic? Or any question from the previous class?
Ma'am, could you tell what is the meaning of orthochromic once again?
Okay. So in orthochromic normoblast, the actually the cytoplasm staining properties change. Okay, so up till this point it is slightly basophilic. Here it is basophilic. Okay, then as hemoglobin starts accumulating in the cytoplasm, so it will also give acidophilic staining. Okay, and here it is acidophilic because ribosome levels is also less, and it has lot of hemoglobin which is present in the cytoplasm. So in the Romanowski staining we have acidophilic stain and basophilic stain. So the depending upon the property of the substance which is present in the cytoplasm, they will take the stain. Okay, some take acidic stain, some will take basic stain. So ribosomes, they will take basic stain, and the hemoglobin is acidophilic in nature. So the staining properties will change, and based on their structure and how they are observed under the microscope, the nomenclature is given. Okay, and this, in this stage of the cell, the nucleus is highly condensed. Okay, so from this cell onward, the cell division is not possible, because the DNA cannot divide, DNA replication is not there, and so the cells cannot divide, and from this stage the nucleus will be expelled out from the cell, and then it will be converted into reticulocyte. So based on their staining properties, their nomenclature is there.
Excuse me, ma'am.
हाँ बोलो.
Ma'am, in this given list, in the left side of the slide, where do we find the colony forming unit and the burst forming unit?
Actually, now this you have to remember both the names. Okay, because in different books we have different names, or you can write the names which are given in Guyton. Okay, you can follow one nomenclature, but you should know what is the alternative name for that cell.
Ma'am, I meant that, ma'am, in the list given in the slide, the left hand side, what can we relate with colony forming unit and burst forming unit? What to relate it with, like common myeloid progenitor or megakaryocyte erythrocyte progenitor, like that?
See, here they have not given the इसके बाद वाली step ना, they have not given. Okay, so these are the common progenitors, और उसके बाद megakaryocyte erythroid progenitor, it will give rise to ये colony forming unit burst, and मतलब burst, एक मिनट, ठीक है. So actually they have मतलब each stage cannot be given. So these are the main progenitors which are involved in erythropoiesis, that is colony forming unit blast or colony forming unit erythrocyte. So these are the which are the mostly which are directly linked with the erythropoiesis. So in that diagram they have not given these progenitors. So these stages are present after that. Okay, erythroid and megakaryocyte colony forming unit, इसके बाद so megakaryocyte erythrocyte progenitor, then it will give rise to that burst form and colony forming unit erythrocyte. So different books give different images and different nomenclature, so you can stick to one, but you should also know what is the alternative name for that cell.
Yes, ma'am. Thank you.
Okay.
Ma'am, in adults, like after 20 or 30 years, the vertebrae and ribs and those bones do the erythropoiesis, right?
Yes.
The graph you showed.
Yes.
But you had also written that membranous bones are the only bones involved, but these are like cartilaginous bones we learnt in anatomy.
So which are the membranous bones?
Membranous bones are cranial vault and some other bones, but vertebrae and ribs are the cartilaginous bones.
So skull is also involved in erythropoiesis.
Could you show the slide saying that membranous bones? I want to, like it was written only or something, then
okay,
there would be a confusion.
Can you see?
So both will be involved in the adults, membranous and cartilaginous both.
Huh. It is not written, only is not written in, mostly we don't write only. ओके ओके, बिकॉज ऐसे होता ही नहीं कि ओनली अभी है कहीं कुछ. It
Yeah,
is
yeah,
not
got it.
mathematics, so we cannot write only or exclusively.
But
ठीक है.
mostly is valid.
हाँ, mostly because sternum, ribs, vertebrae, they are involved in erythropoiesis throughout the life. Okay, so if we want to study bone marrow of any patient, so the most common site from where we collect is the sternum or the hip bone. Okay, from the pelvis, मतलब anterior superior iliac spine and all मतलब from that bone and from sternum we collect the bone marrow if we want to study the bone marrow. If the patient has anemia which is not responding to the standard treatment, then in such conditions we mostly examine the bone marrow. So we collect it from the sternum. So
But in guidance also it is written that in the marrow of membranous bones such as the vertebrae, sternum and ribs. So guidance mentions these three bones as membranous bones. So which is wrong, no?
No, Guyton cannot be wrong. Okay, I need to see for it. I don't know. Actually, I don't remember anatomy. Okay, so what are the answers for the last quiz question? So you have to remember from where we when we ask the patient to do the when we are collecting blood from the patient to for estimation of glucose or sodium, calcium or proteins. So we have to remember from which sample we need. Okay, so suppose we have to check for the glucose of the patient, then you have to collect the blood in the bulb or tube which has EDTA or heparin, so that the plasma can be separated from that blood. Otherwise, if you collect the blood in the plain tube, then it will form the blood clot. Okay, and from that blood clot the serum will ooze out. So serum can, for if you want serum, if you want to estimate sodium or calcium from the serum, then you can collect the blood in the plain test tube. Okay, so that it will give you the serum. But if you want to do hemoglobin, if you want to measure hemoglobin of that patient, hemoglobin level of that patient, then you have to collect it in the EDTA bulb or heparinized bulb. Okay, so for the measurement of hemoglobin, for the measurement of pH or RBC count, you have to collect whole blood, and for all other except for the glucose you have to collect the plasma of the patient, and for sodium, calcium, proteins, and for liver function test, thyroid function test, for potassium or for lipids. Okay, for lipids also we collect the serum. So based on what sample is required, you have to collect the blood in that specific tube. And Aisha,
Plasma for excuse me.
actually the techniques which are used for estimation of blood glucose, they are standardized for the plasma. Okay, so based on the technique and their standardization, we collect the sample. And also if you want to measure clotting factors, then you have to collect the plasma, otherwise the clotting factors will be utilized while the formation of the blood clot. If you collect the serum, then their level will be less in the serum as compared to plasma, because they get utilized while the formation of the blood clot. Okay, so based on the
So why you wrote serum?
what, can you repeat?
Protein, clotting factors and everything will be used up now. So why do we use serum for proteins? We should use plasma, no?
हाँ, plasma protein for
Why plasma? Why not blood? Because cells would also have proteins.
so but we don't want the proteins from the cells, we want proteins only from the plasma, then we have to take only the plasma or serum. Okay, when we measure albumin, globulin, Okay, we don't measure it from the cell, we measure it only in from the plasma portion. So proteins I will check, okay, and I will tell you in the next class, but for all other factors we have to collect the serum. Except for huh?
Ma'am, if sodium is checked in serum and plasma, will the levels be different or same?
Studies are available for calcium, so a lot of difference is present between calcium which is present in the serum and plasma, okay, because calcium is also required for clotting, and the difference is observed in the level of calcium when we measure in the serum and plasma. I am not sure about sodium, but for calcium the difference is present.
And then why do we prefer serum over plasma even though there is a difference?
Because the techniques which are used, they are standardized for that purpose. So for that, I think for that purpose we need to collect the serum. You also search for the for these answers. I'm also searching because these are mostly related to laboratory medicine, and once you enter into the third trimester you will learn in detail about these things. Okay, so Aisha has one question. What is the role of reticuloendothelial cells in regulation of plasma proteins? Aisha is there?
Yes, ma'am.
Okay, so what is your query?
The role of reticular endothelial cell in the reversible equilibrium between plasma protein and tissue protein.
So have you read? It is given in Guyton. So what you have not understood?
I don't understand its role.
Okay. So what is the function of reticuloendothelial cells? They can break down the plasma proteins into amino acid, and they can provide this amino acid to the tissue, okay, for the synthesis of proteins. So what is the meaning of equilibrium? So blood will have either amino acid, okay, or proteins. So these proteins can be broken down into amino acids, and these amino acids can be utilized by the tissue for the formation of its own proteins. So if there is a muscle, so in the muscle the types of proteins which are present is different, okay. So if we have the amino acid in the circulation, it will take the amino acids and it will lead to synthesis of its own proteins. If the plasma protein level decreases, then there will be breakdown of these muscle proteins, and they will be converted into amino acid, and they will enter into the circulation to maintain the osmotic pressure, okay. So that is the equilibrium between the plasma protein and tissue protein, okay. So
Ma'am, there is a diagram in Guyton
yes,
which shows that amino acids are being taken up by reticular endothelial cells.
yes, there is a diagram in the Guyton, and they have given the text for it also, okay. So in the next
Ma'am,
class,
is this right?
yes, it is right. Guyton, in Guyton it is given, then huh.
But then you said na ki they take up proteins and give out amino acids for
So for
to the bloodstream.
So for the maintenance of equilibrium, so the arrow is given on both the directions. Have you seen the diagram? So the arrow indicates that that reaction will take place in both the directions. That's why they maintain the equilibrium. So we will discuss it in the next class also, okay. So I will leave now. So you have next class with Dr. Jarial. So bye bye.
Thank you.
हाँ सर.
दिखा दूँ ना, आपका जो मॉर्निंग लेक्चर है वो मैं ले लूँगा मॉर्निंग टाइम में, बिकॉज मेरे को कल दो लेक्चर है ना आपके भी मेरे भी, मुझे दोपहर में कहीं काम है तो मैं मॉर्निंग में
सर 10 वाला मेरा जो है आप ले
मैं ले लूँगा, मैं इनको बता देता हूँ।
ठीक है सर।
थैंक
Okay, thank
यू
you.
थैंक यू। Good morning, everybody.
Good morning, sir.
Lovely. Okay.
Good morning.
Okay. So we have next lecture today is on body fluids, that is intracellular and extracellular. In brackets, I have written interstitial and intravascular. Okay. So let's do a bit of sanitization of things. That's also required these days. Keyboard and mouse. Okay. So in the last two classes, we discussed the homeostasis and followed by the major role that the autonomic nervous system plays in as a mechanism of homeostasis, and we looked at various pathways from the understanding of the external environment and the internal cues which are taken to the various centers in the brain for behavioral, hormonal and the so-called physiological responses. So we'll now move on to body fluids, and again, like I mentioned, that your medical practice is largely about maintenance of homeostasis, and it's a very common sight in any film that you see a patient and there'll always be an IV line put on and some drip is going on. So effectively what that drip is doing is in some way looking at the body fluids and maintenance of body fluids. Okay, so maintenance of the intracellular environment is cellular property, but the maintenance of the extracellular fluid, which is interstitial and intravascular, is what the homeostasis does. So we will now look at the various compartments of body fluids and how do we measure them in experimental condition, as well as I'll talk about in clinically how do we estimate how much body fluid does a person have. Okay, now a bit of the evolutionary origin, all of you are aware of this, I'm just passing through this. So we understand that we as a multicellular organisms have developed from unicellular organism. Some dates have been estimated about 3 billion years ago, based on fossil records and other inputs. So this is I've taken from one of the textbook, and this shows the how from a unicellular organism as a multicellular organism as human beings we have become. Now the question which I'm putting to you is, what difference is between multicellularity and aggregation of cells? Are the two same? Responses?
Division of labor.
Yes, in a way, but how is the multicellularity different from coordination? Archit, division of labor, somebody mentioned. Perfect, interaction. All correct ideas. So multicellularity is
Another example that we gave, different society of human beings was collection of human beings. So all the same idea that you already mentioned. That let me just expand it a little. Let's see your responses. So integration, coordination, cooperation, communication, fantastic, common function, interaction, coordination. So basically all of you are on the same page as I'm talking about. So what we are looking for is organized cooperativity between specialized cells in terms of structure and function. That's multicellularity. So looking at a little diagrammatic representation of different cell types in different colors. Aggregation is, or multicellularity, without boundaries. So this is a single layer, this is a multi-layered, and there are gaps in between these cells, and it is these gaps which are filled by what we called as the internal environment. And like in the class we discussed earlier, that the constancy of internal environment is something that Claude Bernard emphasized, and later Walter Cannon gave it a name of homeostasis for the mechanisms which maintain this internal environment. Okay, so now what are the challenges of multicellularity? Let us say you do have multicellularity, then what do we need for all these functions to be achieved? And function and organization, of course, structure and function go hand in hand. So you will need a mechanism to deliver oxygen, nutrients to the cells that are away from the external environment. You need mechanisms that hold the cells together. You need mechanisms that allow cells to communicate in between themselves. And so over the time, what is in the red is what is the intravascular compartment. You need fast moving mechanisms because diffusion is not sufficient for a large body for exchange of materials between the cells and transport of materials from the oxygen absorbing area or the nutrient absorbing area. So we have a GIT lined in yellow, exposed on both ends to the external environment. So you need a mechanism that can pick things from there. You have something in the green over here. I'm representing that as a lung. So oxygen and the air would be moved between the two as ventilation, and exchange of gases will occur. Ultimately, it still must reach the internal cell somewhere. So in different colors, we've got different cell aggregates, what we can call as the organ systems. And you have two compartments effectively in between the cell, not so fast moving or very slowly moving interstitial compartment, and a fast moving compartment which is the intravascular compartment or the blood. And the whole cardiovascular system effectively in some way evolved to ensure that the delivery of nutrients, oxygen, carbon dioxide gases, hormones, all methods of communication are passed between these cells very fast. Okay, so again diagrammatically showing intravascular fluid and the interstitial fluid. So we are looking at three compartments now. What is inside the cell as intracellular, in between the cells but not in the vessels is the interstitial fluid, and what is in the blood vessels is the intravascular fluid, and together the interstitial fluid and the intravascular fluid will make up what we call as the extracellular fluid. From the point of view of dynamics, we must understand that the fast moving compartment is in the circulatory system and its integrity is critical for life. So the fluid compartments, three major compartments: intercellular, interstitial, intravascular. These two combined as extracellular fluid. Obviously, apart from these three zones, we have additional fluids in our joints, pleural space, in eyes as vitreous and aqueous humor, in brain as CSF, some fluid in cochlea, and there are multiple places where we have these some amount of fluid. And all these fluid together are called as transcellular fluid, and they amount to about 1 to 2 liters. Okay, so 4 organ systems interact with the environment. Skin, because I mean, even though it directly doesn't absorb anything, but it has a capacity to lose fluid. Gastrointestinal tract in human beings, of course, is for absorption of all nutrients and fluid. Urinary tract doesn't interact environment in that sense of exchange of material, but as a passage, but it still interacts with the environment. And obviously lungs. So the two organ systems, which basically is GIT and lungs, are responsible for all the necessary ingredients for survival, while skin and urinary tract majorly in human beings are for losing material or the waste products. In lower animals, of course, skin is also important for breathing. Now, this is data I have taken. I have forgotten from which book I have taken, but this is the idea. The fluid intake versus fluid losses. Now, obviously, this is an average value, assuming a lot of external conditions are stable. So, if you are in a hot environment, this may not be appropriate. If you are in a very, very cold environment, this may not be appropriate. Also, depending upon availability of fluid in and around you, these values are not sacrosanct, it will vary. But on an average, this is what we generally take. So, as liquid, meaning drinking water, we have about 1 liter. With food, we get some, and metabolic oxidation in some process of biochemistry, you will realize that in the process, water is produced. So, total about 2,500 ml, and then we lose fluid through lungs as we are breathing in. The dry air comes in, gets humidified, goes out. What we call the insensible loss of water. Obviously, skin we lose some amount, feces and urine. These are the places where we lose fluids. So there's all generally a balance between the two, and you have very strong negative feedback mechanism as well as feed forward mechanisms to take care of it. Interestingly, when I was discussing regarding homeostasis with you, and I was telling you that when you lose fluid and the electrolyte concentration increases in your body, there are osmoreceptors which detect this change and initiate negative feedback mechanisms. What has also been found that human beings actually drink water even before the dehydration sets in or the electrolyte concentration increases, meaning that the dryness in our mouth for the urge to drink water is almost like a feed forward mechanisms, even though the negative feedback mechanisms also exist. But in a natural conditions where water is available, freely available, humans do not land up in dehydration. We land up in dehydration simply because we are not carrying water with us, or our environmental and societal conditions are such that we may not be able to have water. For example, when you give your exams, nowadays of course the water bottles are available, but earlier they would, because of security, they won't allow you to take water. So for 4 to 5 hours you would be without water, and you may just become dehydrated. So the idea that the fluid the fluid osmolality as a negative feedback mechanism is true, but also a very robust feed forward mechanism exists which initiates thirst even before the osmolality has begun to change. Okay. So let's come to the total body water and its distribution, certain values. Now, whether you like it or not, this is something that you have to unfortunately remember, because these are the kind of questions the entrance examinations and other exam conducting body during exam viva classes, these are generally asked by faculty because it's easy to ask these questions. They are the values, and you will have to remember them. There is no option. So total body water is 60% of body weight. Now obviously this assumes certain conditions that you are not either overtly fat or very thin. It assumes a mesomorphic body type where 60% of body weight is water in liters, and of which 2/3 is the intracellular and 1/3 is in the extracellular. So these are the 40-60% divisions. The interstitial is the 3/4 of the extracellular fluid and the intravascular fluid. Certain values are given as 28 liters, 10.5 liters, 3.5 liters. Now kindly note that in the intravascular fluid, the blood volume is 5 liters, but the fluid, which is plasma, is about 3.5 liters. The rest is RBC. So what is there in the RBC is not considered as the intravascular volume of the blood. So the cellular compartment, cellular composition of the blood is about 40% to 45%, as the cell RBC and WBC very small amount, and the rest plasma is about 3.5 liters. So trick questions are generally these, that the intravascular fluid volume is, so you should not write 5, it is actually 3.5. Below I will also mention the protein, minerals and fat percentages. These are taken from Ganong. So again, different textbooks have different percentages mentioned, but this is just a general idea that you should be aware of. On an average, this is the kind of values that we have in our body. So total body water is about 42 liters, 28 liters within the cell, 10.5 liter in between the cell, outside the extravascular fluid at 3.5. intravascular fluid. Now, from medical perspective, when you lose water, you will lose actually from all compartments sooner or later. So even if I lose water from intravascular fluid, the water will shift from interstitial to intravascular fluid, which will then again shift from intercellular to the interstitial fluid. So over a period of time, they will all equalize in terms of the distribution losses. So percentage loss in one compartment will manifest into the other compartments as well. However, your body systems are designed to hold two things. One, you have cellular systems which manage the intracellular fluid volume. Each cell has its own mechanism. Someday perhaps you will learn about that. Today I am focusing only on as a homeostasis we focus only on the intravascular and interstitial compartment. So when you begin to lose fluid from the extracellular fluid, the major threat is to the cardiovascular system, because if the blood pressure drops, you die. Okay, so I may be able to lose an interstitial fluid for about 2 liters, nothing will happen to me. But if I lose 2 liters from the intravascular fluid, I'll die. And our medical management primarily looks at the intravascular fluid volumes. Of course, in diseases, the interstitial fluid also increases or decreases, manifesting as edema or the tissue dehydration. But so when we put an IV line or IV drip, we are basically putting stuff into the, yes, somebody's, yes, Jaysimon, please. Yeah, something Jaysimon was asking. 18%.
What is the meaning of protein is equal to 18%, mineral is equal to 7%? What does those mean? What do they mean?
In weight by weight. Weight by weight. So it will be.
Sir?
Yeah.
Wait, no. Okay.
18. So somebody is
Sir,
100.
and another thing.
That's correct. Mohit mentioned.
And interstitial is equal to 3/4 of ECF, and in brackets it's given 15%. So
Yeah.
that 15% and this 5% will together make 20%.
Correct.
That is ECF. Does
Right.
it mean like that?
Yes, it does mean like that. So this 40, 15, 20 is basically looking at percentage of the total body weight. See, it's basically which fraction do you want to see? I mean,
I can understand, sir. I can get it totally.
yeah. So I mean, it's a different way of looking at it. So if, I could say that.
Thank you, sir.
Yeah. So the interstitial fluid is 15% of total body weight. That's one way of putting it. Or I could say it is 75% of the extracellular fluid. Same thing. Okay. So it's more of a trickery of mathematics. More importantly, the absolute volumes will matter. And see, in actual clinical practice, you're not going to measure ICF, you're not going to measure interstitial fluid. All that you're going to do is look at the intravascular fluid in your medical management. All your life practices, this is what you'll be doing. Okay. So, but these are the information that you need to have at the back of your mind as to understanding the physiological basis of whatever you're doing in medical practice. All right. And the transcellular fluid, synovial fluid, pericardial fluid, and all other fluids, joint fluid, everything is about 1 to 2 liters. That's the estimated values. And in disease condition, mind you, the transcellular fluids can become very large. So I can have a pneumonia, which leading to large amount of fluid in my pleural space, or I may have liver cirrhosis, which may manifest as ascites with large amount of fluid in the peritoneal space. So those are the conditions which occur, and maybe in lectures in the microcirculation that you'll be taught under cardiovascular system, you will touch upon these pathologies where the transcellular fluid becomes huge, large amount. Okay, so this is what I was talking about, that loss or gain of free water in any compartment affects distribution across the compartment. I mean, understandably because of osmosis. However, the intercellular fluid and the interstitial fluid, you have a cell membrane, and it has active machinery. It has active machinery to hold its own water. So even when you begin to lose interstitial fluid or intravascular fluid, that is the extracellular fluid, a cell will try to maintain its volume. However, there is a limit to it, and ultimately it will lose water. And clinically, it's nearly impossible for us to look at the intercellular fluid We majorly look at the interstitial fluid as the turgidity of the skin or sunkenness of the eyes, and we make a judgment as to the interstitial fluid. Or we take blood pressure and see, okay, the blood pressure is very low, and we assume that it is because of the low intravascular volume, because you need volume for pressure to develop. So in this way we estimate clinically. But otherwise, from the perspective of physics, this is what it is all about, that the fluid will move across. Now, again, just reiteration of what you already know perhaps, is when you have a semi-permeable membrane with solutes distributed differentially and solutes cannot pass through semi-permeable membrane, the solvent moves towards the concentration side and what we call as the osmotic pressure. And by definition, a pressure which is required to be put on the more concentrated side to prevent osmosis of water would be called as the osmotic pressure, and this is the formula which computes it. I do not know how they have computed, I have forgotten actually how they come to 19.25, but that's the equation to compute. So theoretically, now if you look at the osmolality of the blood plasma, it's about 300 milliosmoles. Okay, so if you multiply 300 with 20 round figure, you'll get about 6,000 millimeter of mercury, which is huge pressure. So that is the pressure if you were to take plasma and put it against distilled water with a semi-permeable membrane which allows nothing to move across, nothing, meaning not even salt or proteins, nothing. That's the kind of pressure it has. However, in the body, when you look the compartmentalization between the, let me take the interstitial fluid and the intravascular fluid. At the capillary level, everything can move in and out. What cannot move in and out is the proteins in the blood. So effectively, it is the protein concentration in the blood which acts like an osmotic force, and two osmotic force, and therefore the protein drag which pulls the water into the capillaries is not the total osmolality of the water of the plasma, but the osmolality of the component which does not go across. Okay, so this is an important concept to remember, that while the absolute osmolality, if you take that and compute the osmotic pressure, it will be huge for interstitial fluid as well as for the intravascular fluid. But because the effective osmolality which leads to movement of water is because of the non-diffusible components of the non-diffusible solutes. And between interstitial and intravascular fluid, the sodium, which is the largest ion along with chloride and bicarbonate, they can move easily. They can easily move across. Okay, so two terms you may come across repeatedly in your medical literature: isosmolality and isotonicity. I'm typing it down. So isotonicity, isosmotic और isotonic, isotonic कर लेता हूँ, isoosmotic. Okay, the two are different. Okay, so in terms of physics, both are same, but isotonicity is to be spoken whenever you look at something separated by a membrane. So I can have a milliosmol, 100 milliosmol of glucose, and on the other side I have in another beaker 100 milliosmols of urea, and in other I have 100 milliosmols of sodium chloride. And if you ask me, are they isoosmotic? Yes, they are isoosmotic. All of them have the same osmolality, same number of solutes in the compartment. But if the next question is, are they isotonic? Then you have to ask yourself question: if I were to put this material outside this cell, will it lead to movement of water from the cell? Okay, yes, please, Mohit, go ahead, tell me. Yeah, okay, you are answering, right? So they are so isotonic is always with reference to the plasma, and isoosmotic doesn't matter. You don't look at the plasma, you just look at the the solutes, irrespective of whether they can go across the plasma membrane or not. Okay, so now for example, if you place a cell in a glucose, yeah, so when see, of course, NaCl will diffuse. So when I say 140 milliequivalents is sodium in plasma, the rest is balanced by chloride and bicarbonate and other anions. So 100 milliosmoles of NaCl means dissociated ions. So to get 100 milliosmoles, I have to put 50 milliosmoles in 1 liter. Okay. So in that sense, you are correct that NaCl will be 2 times if the calculation, if your starting point is different, and assuming that there is 100% dissociation. In that sense, it is correct. So the concept which I want you to remember is this: isotonic versus isoosmotic. Okay. Now let me put a question to you. Now the osmolality of plasma is about 280. I'm taking 300 as a round figure, just for sake of calculation. Okay, it's actually about 270 to 280 or maybe 290 in some, varies in people, but this is the value. Let's start with this. Okay, now which is equal to 0.9% NaCl. Whenever we use this kind of phraseology, 0.9% NaCl, what does that mean? That gram in 100 ml. No, I mean I'm just coming to, that's correct. Sushmita, what you're saying is correct. So, but then it is that gram in 100 ml. Okay, so when I say my HB is 16%, we mean 16 gram per 100 ml of blood. Now this is because, perfect. So right, Ishita. So this is a sort of convention. I mean, you could say, well, why all this? That is why the SI system and Americans books will not go by these methods. They use more standardized way of putting at it, while the British books would generally keep to the 16 grams. So you will find diabetes. What is normal blood glucose level? So the someone would say 100 milligram %. This means 100 milligram in 100 ml of blood. Okay. Now another way of looking would be 5 millimoles glucose. It is the same, right? So milligram per DL, that's correct. So if you don't qualify, a percentage means that many milligram in 100 ml. Okay, you could find out the solute's molecular weight and do the calculations. So 5 millimole solution of glucose is same as 5% solution of glucose, almost. Sorry, is same as 100 milligram % of glucose. Same. Similarly, 5% glucose is almost equal to 0.9% NaCl, which is almost equal to around 300 milliosmol. Not exactly, but nearby. Okay. Now, glucose can go through the cell wall. It can be absorbed. So if I now let us look at 3 solutions: 5% glucose, 0.9% NaCl, and your plasma. If the question is asked, which of the two 5% glucose, I forgot the 5%, 5% glucose or 0.9% NaCl is isotonic to plasma? Answer. Correct. Okay. But all three are isoosmotic. Okay. So when so normally if you look at of course you will be seeing when you see wards all the drips that we give to patients when anybody comes and we put an IV line, let's say give them saline, so we infuse saline. Okay, that is 0.9% NaCl equivalent to isoosmotic plasma, which therefore is isotonic. However, if I gave somebody 5% glucose as dextrose, we give 5% dextrose, okay, then I am infusing an isoosmotic solution which will however be not isotonic, and therefore after some time it will be almost like putting the cell into a hypotonic solution. So if you have a patient, if you give 0.9% NaCl, the tonicity of the blood will be maintained. But if you gave 5% glucose, glucose will be taken up and all will be left with water, and actually will make the person hypotonic. Am I okay with this understanding? Therefore, when you want to give glucose, better give it as 5% DNS, okay, which means 5% dextrose in 0.9% NaCl. Am I clear with this? So when you actually give 5% DNS as an IV fluid, you are actually giving twice the isoosmotic fluid as compared to plasma. But because over time the glucose will be taken up by the cells, all that will remain would be 0.9% NaCl, which will be isotonic. Alright? Some response for someone? Okay, done. Ishita. Yes, we need glucose. If person is ill, not eating food, glucose level drops. Pankaj, I'll repeat again. So, when a person is not well in the ward, in the ICU, not able to eat, we need to give him person some energy and also replace fluid. What to give? Okay, person needs glucose and person needs fluid. Now the best fluid to give obviously is isotonic fluid. So we give NaCl as 0.9% NaCl. By 0.9%? Because 0.9% if you compute would be 900 milligram sodium chloride in 100 ml of water. Okay, so let me start all over again if it is okay. No, it is not necessary, Kartik. It is not necessary. Kartik, so that you know, Kartik, so that you know how much we are giving, because you can't overload and you can't undergive. So you need to be aware of how much we are giving. So when I start a drip of 5% dextrose, I know how much glucose has been given to the person in that sense, because dosage matters. Sometimes we also give 20% dextrose when you want to go very fast. Okay. So I'll repeat the whole thing again just to be clear, because this is what your medical practice is all about. So this is where I was speaking about. So units for substances, you all know about that. So 1 mole of glucose and all that. So the calculation is this: 90 milligram glucose, about I've taken the almost equal to 5, about 100. So it's actually 5.5 millimoles of equivalent to 100 milligram percent. The same calculations. Okay. So well, like somebody mentioned that when I take 0.9% NaCl, I'm actually giving 154 millimole. But because it breaks into 2, it becomes 300. Okay. While 5% dextrose is about 185 millimolar. in that sense. Okay. So molar solutions, this has already been touched by one of you. So just to repeat, isotonic solutions are also isoosmotic, but not all isoosmotic solutions are isotonic, because isotonicity is in relation to plasma and cell membrane. Okay, so if a cell is placed in a solution of glucose oblique urea of 300 milliosmol per liter, then it is almost like placing the cell in hypotonic solution, because both glucose and urea will immediately go across the cell membrane. They can pass through very easily. Okay, but if you place it in I may need a correction of 300 milliosmols. Typingly I did not complete that. 300 milliosmols per liter. Okay, 300. So if I place a cell in a solution of 300 milliosmol of NaCl, then I am placing cell in an isotonic solution. If I place a cell in 300 milliosmol of urea, then it is not isotonic, but it is isoosmotic. Okay, so when say somebody is having a renal failure and the blood urea level is rising, if you measure osmolality, it will be very high, but it won't cause shrinkage of cells. Okay, but if the same high osmolality was because of NaCl, the cells will shrink because they are in a hypertonic solution now. Can I move forward on this? Right, thank you. I suggest check up any website, just Google up isotonic versus isoosmotic and you'll get the gist. So right, so moving on to the next component. Again, this picture I've taken from one of the textbooks, but I think most likely Ganong. I prefer taking the values from Ganong and to give it to you, because many of you may give US MED licensing examination and they tend to refer to this book for their values. So it's a distribution of various components within the cells. So you have NaCl, you have to remember all this, unfortunately. So you have to remember what are the major cations in plasma, what are the major cations in the interstitial fluid and the intracellular fluid. Kindly note that in the plasma, NaCl is the major component along with bicarbonate as the osmolality producing solutes. In the interstitial fluid, it is almost the same except for the proteins which don't cross the capillary membrane. Therefore, the protein is absent from the interstitial fluid. This is a very minor amount, but effectively negligible. Then you look at the intracellular fluid. There the condition reverse. Potassium is the major ion along with phosphates and proteins inside the cell. There is no option but to remember this. There is no logic here. Just remember it. So all compartments are isoosmotic. The value 295, again books will vary from 270 to 290 to up to 300. The pH is very narrow range, 7.36, 7.44. So distribution of electrolytes, non-electrolyte across cell membrane. Again, values taken from an average healthy person. There is no option but to remember all these values. Even if you don't remember now, when you will do medical practice, it will become, it will come. But fortunately, the medical practice, whenever you get a report, there's always in the bracket mentioned the normal ranges. So even if you don't remember, the people understand that and they give you the normal values as a reference values in every lab report. And with when you see it hundreds of times, you don't have to remember, it becomes natural. Another concept that you must understand, and it will be again utilized in your clinical practice, is anion gap. So what we understand is that sodium is balanced by chloride and bicarbonate in the ECF. I'm going back. So here you see on the left side the plasma, the major cation is sodium and is balanced by chloride and the bicarbonate. On the left side you see the plasma component, including and also the interstitial fluid. However, in some conditions, so how do we measure? So if I want to see, I take a blood sample of a person, okay? So I measure his sodium, measure the chloride and measure the bicarbonate. Obviously what I'm not measuring is the protein components, and there could be other anions, maybe small amount of phosphate, maybe small amount of other amino acids or fatty acids which I have not measured, but they are also balancing the sodium, so which I am not measuring. So this a gap always occurs. So this gap refers to all the other anions which are present in the plasma or the interstitial fluid, which are balancing the sodium, called anion gap. And if this gap becomes larger, more than 18 to 16, it means that now you have in your blood anions which are not chloride or bicarbonate, something else is there, okay? And these are lactates, acetoacetates, salicylates. And where do these come from? Lactate, where does it come from? Acetoacetate, where does it come from? It is part of the biochemical processes. So when you have hypoxia, tissue hypoxia, lactate will be produced. When you have high amount of lipolysis, because you have not eaten for a very long time, you are hungry, the fat breakdown occurs, ketone bodies, correct? Chaitanya, that's correct. So whenever you are hungry, and the acetoacetate will go up, and therefore you will have larger anion gap. So if I really wanted to find out, so if I take a blood sample of a person, and as a patient is there, and if I find a very high anion gap, I must immediately look what is that is increasing. Is it the drug which I am giving, like salicylates, or the drugs that you give? Is it there's a tissue hypoxia which is leading to production of lactate, or there is very high lipolysis? That means I must give glucose to this person. So utilizing this, while you may not get the exact answer as to what is causing the anion gap, at least it makes you think about it. So anion gap refers to the unknown anion which is balancing the sodium. Am I okay with this idea in medical practice? Yeah, lovely. So now how do we measure? So again, I like to put everything in the patient perspective, because that's where our whole life is going to be. So I have a patient in the bed, lying on the bed, and somebody asks me, can you tell me how much is the patient's total body water? How much is patient's intravascular fluid volume? How much is the patient's interstitial fluid volume? I mean, we would love to know it. Unfortunately, in regular medical practice, we don't do any of this which I'm going to teach you. This is primarily more as a research tool, because it is so cumbersome to measure that, and perhaps your utility is not that very high to mandate its routine use in patient management. Therefore, we don't use these techniques for actual patient care, but hopefully with improvement technology, better chemicals, we may come to this stage as well later in life, later in medical development, but not as of now. So what do we do is, we take a known substance, put that substance into unknown volume of water or fluid, look at the dilution, do the calculations, okay? What do you call it? Volume or distribution, or volume of distribution, or volume space, okay? A terminology you will come across. So I may say that this drug which I give to the patient has volume distribution of 20 liters. It basically means out of 42 liters of the total body water, this drug diffuses only into the 20 liter space. It doesn't diffuse throughout the body. And this is useful from when the drugs are being developed. They do all these things, because they need to understand that how much is the dosage required for the drug to reach tissues or to the synovial fluids, or whether drug actually reaches the cell, or drug stays only in the vascular compartment, or the drug moves only in the interstitial compartment. So they measure all these things. Some drugs are lipid soluble, so they find out this volume of distribution. So I give a drug and my volume of distribution is 50 liters. It basically means this drug not only goes into the fluid space, but also goes into the lipid space. So in that sense, you can compute, or you calculate, or make drug dosages when you design drug therapy. So this, of course, this is not our job. This is done by the expert pharmacologist, clinical pharmacologist. As medical doctors, we just follow the orders. Okay, Sagar. So let us say if I had a drum full of water, and somebody asked me to tell me how much volume is there. One, you could use physics, and you could take length, breadth, and diameters and do the calculation. But if, let us say, that was not allowed, you had to measure it, not calculate it. So there are three things. I'm typing it down. Please note. Calculate, measure, estimate. What is the difference? Calculate, measure, estimate. Okay. So if I had a drum full of water, I could calculate its volume by formula, or I could take a measuring cylinder, transfer all the water into measuring cylinder and actually measure it, or I could put a small amount of a coloring agent or small amount of any solute which I know, known volume, and drop it. After some time, take a sample and measure the dilution, and then I will use a formula. I put 10 gram of solute, any solute substance, I put that into the drum. So I'm putting 10 gram. After a while, when it is all equalized, I take a sample and I find it is 1 gram per liter. So how many liters was there in that beaker or something? Absolutely 10. So this is a dilution method, right? Now there are certain assumptions that the substance per se does not cause fluid shifts between the compartment. The substance is inert, it's not metabolized. So if I use glucose to measure, assuming glucose will go everywhere, but then what will happen? The glucose will vanish because cell will use it. So I will overestimate. So, and the substance doesn't cross the other compartment. So I'll go to another slide. Next slide. This is again, I think I taken from Genong itself. Various indicators which we utilize for experimental purposes. So if I want to measure total body water, I use tritiated water or antipyrine drug, give it to a person, wait for some time. This antipyrine and water will go across all over the body. Take a sample from blood, do the dilution principle, you get total body water. If I want to find extracellular fluid, I will give sodium isotope. Give it. I assume that sodium will not go into the cell because all sodium is outside. Therefore, wherever sodium will go will be either the intravascular compartment or the interstitial compartment. Do the dilution principle. Their assumption, but mind you, sodium does go inside. So it will be rather than an estimate. We don't measure it, it is estimation. So all these, though we write measurement of body fluid, is actually an estimation. We are not actually measuring it. So plasma volume, similarly blood volume, interstitial fluid. Okay. Now we do not have a compound which stays only within the interstitial fluid. Though we can't measure interstitial fluid volume. We actually calculate it by total ECF minus plasma volume. So it's just a calculation. In that way we find out various volume. Similarly, intracellular fluid. I mean, I do not have a agent which stays only in the cell and not in the ECF in the ECF. So if I wanted to find out intracellular fluid volume, I will first find out total body volume, total body water, then find out ECF and then subtract it. So I mean, you need to have a compound which stays within that compartment to be able to estimate the volume of that compartment. This particular table, please remember them, because in your examinations, MCQs, faculty and staff generally have want to ask you these questions. But like I mentioned earlier, these are something that we utilize only in research laboratories. When you will be seeing patients, we won't be using any of this except for maybe blood volume, but that too not by chromium labeled RBCs, mainly by the formula which is given below, which is given blood volume is equal to plasma volume minus 1 upon hematocrit. So in clinical practice, we do not do any of this. Okay, but as a principle, you should be aware of these. The red arrow points out to the compartment which are calculated, not estimated. Okay, certain information. With age, we have decrease in water content, increase in fat is decrease in water content, and the lean body is these values. Okay, so therefore it becomes actually 60% when you factor in the fat. These are important because old people who are old can get into dehydration faster than a healthy young person. Okay, so the homeostatic mechanisms to defend water, body water. Sensors we discussed last time. We have ECF sensors which are present in the brain as the organum vasculosum of lamina terminalis, OVLT, CBO organs we spoke of. So volume can be sensed, pressure can be sensed. Effectors are thirst, kidney, skin and hormones. Integrators are ANS, hypothalamus and limbic nervous system. So redistribution. No matter which compartment loses water first, eventually all compartments will lose water first. Isoosmolarity oblique isotonicity. And this is what we do in medical practice. When we have diarrhea, which compartment loses fluid? Can you tell me? In diarrhea, which compartment loses fluid first? ECF, which part of ECF? Intravascular or interstitial? Interstitial. So because first interstitial goes, and then it manifests as the intravascular loss. When you interstitial, it is interstitial, Anaga, and the others interstitial. Okay. And when you lose large amount of interstitial, you will lose IV also. Perfect. Therefore, when you have severe diarrhea, initially, sunken eyes, cheek, everything will be inside, low turgor, lose large amount of fluid, BP will go down. But when you have burns, okay, if you remember, somebody has, let us say, hemorrhage, then obviously IV. Okay, perfect. So, but if you lose blood slowly, then the fluid will move from interstitial to intravascular, and you will be able to handle large blood losses, slow large blood losses over time. Yes. Okay. So initially, obviously, the insult is to one compartment. If it is slow, other compartments are able to balance it out. If it is too fast, especially intravascular, it's a medical emergency. How to treat? What to do? How to treat? You have diarrhea and vomiting. Yeah, but to stop bleeding, you could give ORS, but let us say that there is too much of vomiting. Person can't absorb. IV fluids. Yeah. Okay. So preferably give DNS, dextrose normal saline. Okay. You could give normal saline also. Normal saline meaning 0.9% NaCl. It's called normal saline. NaCl is normal, is NS. Normal saline, dextrose, normal saline, DNS. So 5% DNS, okay, or So depending upon what are the conditions whether the person can take orally or person cannot take orally, you give add ORS to this. Okay, so this is what I had to. Yeah, there are. So you have IV fluids for which contain nutrients. Okay, you have a person cannot take anything from by mouth. So your total what is called as TPN. Okay, total parenteral nutrition. Spellings. Okay, so you give TPN. So person can't take anything by mouth, so you have to give it IV, all the nutrients. So there's a combination of that. You give plasma, you give platelets, you give RBCs as separate things. So they're all effectively IV, but you don't call them IV fluids. So you give dextran. Okay. Dextran for proteins. It's an example because somebody I saw osmotic fluid with 20% albumin to keep the proteins high. Proteins for plasma protein component. Okay. So I'm just giving you a list. Obviously, the first everybody starts with normal saline or DNS. If you take blood samples, do lab investigations, find okay the protein is also low. Let me give some protein. So accordingly, you will change. Now, let me ask you a question before I leave. I don't know who has the next. Just I have to take this call. Just to replace if somebody can have. I'll just hold on. Yeah. So see, if somebody is having diarrhea, obviously the person has lost salt and water. So you want to give water. By giving whole salt diet, person will consume water. So it doesn't mean withholding water. So high salt diet doesn't mean withhold water. So somebody take high salt diet, automatically will take water. I just take this call. Manoj. Oh yeah, Sagar, I will do that. I've been just quite occupied with the exam section, so I have not been able to do that. Any queries on this? So because I'll have to leave now. That amount of water loss is very minimal. See, your for water loss through purgatives is how much? 200-300 you will have water. No, salt means regular salt, NaCl, not magnesium salts. This I'm talking about when I say high salt, it means NaCl. So when you give purgatives, you lose maybe 200, 300, 400 ml of 500 ml of water that you will drink, so it will be easily compensated. No, not a problem. The problem occurs when you have diarrhea and you are not able to hold water. Within diarrhea, you may, if you go to washroom what we are looking is sodium salts. Because you don't want to increase magnesium level in your blood. You want to maintain NaCl in your blood. So you won't give magnesium salts for maintaining osmolality of blood or plasma. Alright, any queries for today? Tomorrow I have 2 lectures along with Dr. Gitanjali has 2 lectures. So, but I will be taking the morning lectures because I have some work in the afternoon. So, I'll take my lectures in the morning. So, I think we can continue again around about this tomorrow. So, you can read up from wherever you want. What is IG? Pankaj, I couldn't understand. IG? Yeah. We'll have a look at this tomorrow. So I suggest read up from wherever you want. If you have any queries, we'll take up tomorrow. Okay. IG means okay. Thank you for the language update. Okay, my daughter also perhaps understands all your terminologies. All right, bye. हाँ मनोज आ गया. हाँ मनोज आ गया. Hello, Manoj. Hello.
ठीक ठीक। तो बस
अरे मैं ये कह रहा था किस नाम से आएगा? डॉक्टर सुदीप, आगे क्या है? नहीं, दो लड़के आ रहे हैं, एक तो आ रहा है आदित्य थकरे। अच्छा, डॉक्टर आएगा, डॉक्टर सुदीप ऐसे लिखा आएगा? सुदीप सेन, ठीक है, चलो चेक कर लेता हूँ। ये तो दो ही लड़के हैं, क्योंकि इसमें जाके करना है ना, पीपल में जो ये पीपल में जाके 4 लड़के आ गए, एक और आ गया। अच्छा, तो यहाँ ऑन रखो, यहाँ माइक बंद है। अच्छा, 2 बजने में 5 मिनट हो तब कर दूं? हाँ ऑन रखूँगा। और कैमरा तो नहीं रखना ऑन? कैमरा वीडियो जो है यहाँ पे एक साइड यार टर्न ऑन कर दूँ? ऑन भी कर दिया। और ये माइक रहने दो बंद, माइक बंद रहने देता हूँ अभी। ठीक। हेलो सर, नमस्कार। हाँ सर, वो सामान आ गया क्या? सामान आ गया क्या सर? अच्छा, ठीक 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 है सर। और दूसरा सर वो, हाँ और सर वो करानी है आपने सेंड। अच्छा अच्छा सर, करा देना प्लीज़। ठीक। हाँ भैया। अरे मैंने ना अभी सुदीप सर को प्रेजेंटर बना दिया है। हेलो।
Hi, good afternoon.
ठीक है ना?
Am I audible? Are my slides visible?
हाँ मतलब, रुक जाओ, रुको रुको रुको।
Okay, thank you.
हाँ चल गया चल गया। ठीक है, मैंने अपना माइक और कैमरा बंद कर दिया है। हाँ ठीक। और प्रेजेंटर भी बना दिया सर को ना। जी सर।
सर का क्लास है अभी।
हाँ कर दिया, शुरू हो गया।
सर को प्रेजेंटर बना दिया।
हाँ कर दिया।
ठीक है।
ओके, चल रहा है जैसे
हाँ,
ना
ठीक है।
सेंसर ना। कर दिया।
थैंक्स।
कर दिया। वो तो सर कह भी रहे ना सर, वो कह रहे हैं कि मुझे समझा रहे सभी लोग, तो ये तो क्लास टेस्ट में देखना चाहो तो देख लो 1 मिनट।
नहीं ठीक है, हम इधर ही आएंगे, इधर एग्जाम देने का फर्स्ट ईयर का।
कब?
अभी 1 घंटे के लिए।
नहीं अभी नहीं ना, 3 बजे से होगा ना।
नहीं नहीं, 3 बजे से दूसरा लेक्चर है, बायोकेमिस्ट्री का ही है। वो भी एक्चुअली है ना, LT1 और 3 में एग्जाम के लिए बुक किया हुआ है।
हाँ।
एक मिनट। तो अभी तक 2 से 4 बजे 2 लेक्चर बायोकेमिस्ट्री का ही है।
तो बच्चे
2 से 4 बजे का जो वो है ना, हाँ 2 से 4 बजे का जो लेक्चर है ना बायोकेमिस्ट्री का ही है।
हाँ।
दोनों अपने घर से ही ले रहे हैं।
अच्छा मतलब,
एक सुदीप सर 3 बजे तक है, इसके बाद अल्पना है, वो भी अपने ऑफिस से ले रहे हैं।
अच्छा।
तो ये LT 4 बजे तक फ्री है।
हाँ।
तो 1 से 3 में जो एग्ज़ाम था, कुछ स्टूडेंट को हम इधर भी बैठा रहे हैं।
1 से 3 बजे तक।
हाँ नहीं, 1 और 3 में जो एग्ज़ाम अभी
अच्छा।
होगा,
वो यहाँ होगा।
हाँ वो इधर भी होगा क्योंकि थोड़ा कम भी
नीचे
होगा।
बुकिंग करा दी?
नीचे 1 और 3 का बुकिंग कराया था।
नहीं नहीं, टू का करा दिया?
टू का नहीं कराया है।
अच्छा, करा क्या है?
अभी टू का तो ऑफिशियली पेपर में बुक नहीं कर सकते ना, क्योंकि क्लासेस चल रहा है ना।
हाँ तो नहीं होगी ना फिर।
तो वो इसीलिए कि वो बायोकेमिस्ट्री का एग्जाम चल रहा है। तो हमने दोनों फैकल्टी को बोला है, तो ये लोग घर से ले रहे हैं।
मतलब आपको मन हो रहा है ना सर?
मैं बोल दूंगा नीचे जाकर, राजीव जी से बोल दूंगा एक बार, क्योंकि एक्चुअली बुक किया हुआ था 1 और 3 है एग्जाम के लिए, और ये यूजी लेक्चर के लिए था। तो बस अभी ये दोनों 4 बजे तक फ्री है, इसीलिए हम सोचे कि कुछ स्टूडेंट्स
केमिस्ट्री, बायोकेमिस्ट्री से
बायोकेमिस्ट्री से ही है। मैं सर हूँ बायोकेमिस्ट्री में।
बात कर लो, मन से बात कर लो। अरे ये दिक्कत हो रही है कि हमारे कंप्यूटर में भी आवाज़ आ रही है। अच्छा दूसरा ये बायोकेमिस्ट्री से ऐसा रहा है कोई, और ये कह रहे हैं 2 से 4 बजे तक एग्ज़ाम यहाँ कराना है।
मनमोहन जी है क्या? मैं बात करता हूँ।
लो बात कर लो इससे ग्राउंड।
मनमोहन जी, मैं वो बोल रहा था कि सुदीप सर का लेक्चर है ना, अल्पना मैम का है, बायोकेमिस्ट्री का ही है। मैम ऑफिस से ले रहा है, वो भी खाली है, इसीलिए हमने मैम को सर को बोला है। तो फिर नहीं क्लास में कोई प्रॉब्लम नहीं होगा। 3 से 4 के लिए भी मैं ही वो इधर पैसेंजर वो कर दूंगा मैम को, फिर वो मैं कर दूंगा। मैं इधर ही हूँ पूरा टाइम। ठीक है ठीक है।
अरे 4 बजे तक रहेंगे ना, 4 बजे से ज़्यादा तो नहीं रहेंगे ना?
क्या अच्छा
हाँ
नहीं 4 से ज़्यादा नहीं, 2:30 से 3:30 तक ही है बस।
अच्छा
उसके बाद
ठीक है,
करेंगे।
ठीक है। मतलब मेरे को प्रॉब्लम नहीं है, आप लोगों को प्रॉब्लम नहीं होती, डिस्टर्ब नहीं होगा ना?
नहीं ये कुछ भी नहीं होगा। बच्चे आपके एग्जाम देके चले जाएंगे। 1 घंटे का एग्जाम है, 2 घंटे इसमें हमें कुछ नहीं देखना है ना। 3 बजे तक तो ये सब
कुछ नहीं करना। आवाज़ ऐसी आएगी, क्लास चल रही है तो आवाज़ आएगी, उसको छेड़ना नहीं है। क्लास चल रही है ना? इसके बाद कम अम कुछ नहीं होगा सर, इसको छेड़ना मत।
हाँ नहीं कर रहा हूँ कुछ भी। नहीं मैं मुझे लग रहा है कि किसी ने वो नहीं किया होगा ना, इसीलिए।
नहीं वो होगा ही, वो दूसरे लोग के ना, हम
ठीक है।
वो उनके हाथ में है अब।
हाँ वही है, सुदीप सर का ही आवाज़ होगा मोस्टली। ठीक है अभी कुछ नहीं कर रहा हूँ। क्या मैं तो सर को बोल तो नहीं रहा ना। ठीक है ये तो सर वेट ही कर रहा लग रहा है, 2 बजा नहीं है ना।
वेट करें ना सर।
Good afternoon. Am I audible? And are the slides visible?
Yes, sir, you are audible and they are visible also.
Thank you. Good afternoon, everybody. So, I'll again start off with a fresh topic today. There are some small areas which are left over, and we'll probably touch upon those areas on Monday. There are a few things in protein structure which are left over, and a few things in amino acids which are left over, but I'll probably take those leftover areas on Monday and start off with separation of proteins today. If any of you have any doubts, please ask, otherwise we'll proceed. Could you read or could you go through the topics? I've sent you the slides. I hope everybody has received the slides. Good afternoon. I sent it on the common email. Please go through the topics. These are new topics, and if you have any doubts, please ask. So I'll be again joining you on Monday, 21st of December at 10 a.m. I have another class. So what I just explained is I'll be starting off with a fresh topic today, separation of proteins, which was scheduled for Monday. So I'm taking that separation of proteins today because some conceptual things need to be clarified, and then we'll probably finish up some remaining areas on Monday. Okay? So if you don't have any questions, I'll start off with separation of proteins. This was partly covered by Dr. I'm forgetting his name, Dr. Asta. Yeah. So Dr. Asta covered this in her practical class on Monday afternoon, where she talked about protein fractionation. Yeah, we'll talk about structural things. Again, I don't want to go into mucoproteins and glycoproteins right now because it will take a little time. So we'll talk about some structural clarifications on Monday. Okay. So just to continue, I would like to ask, please use the chat box, why do you need to separate proteins, or why do you need to purify proteins? This was already covered in your practical class, but I'll start from there. Why do you need to purify proteins? Why do you need to separate proteins? For example, when we do a analysis in serum, okay, structure and function. But as doctors, why do you need to know about structure and function? You are not scientists. You are going to be doctors. So why do you need to know about structure and function of a protein? A scientist will need to know that. As a doctor, why do you need to know about a structure and a function? To isolate protein of interest, okay. Hormones, okay. Diagnosis. So, say for example, for the sake of argument, when you do a blood analysis, you still know albumin is high or low and globulin is high or low or some particular protein. You don't need to separate it out. So when you talk about analysis, I do a blood analysis and I know whether serum proteins are increased or decreased, and why do I need to purify proteins? Why do I need to go into all these complexities, all this chemistry and complex concepts? As a doctor, if I'm aware of the levels, that is all I need to be worried about. So why do I need to purify proteins? Why do I need to separate proteins? Chat box, diagnosis, okay. I would have loved to be there physically, but unfortunately we'll have to do with the chat box. Okay, you guys are right that you will be knowing more about the structure and function, but my question was more specific, that as doctors, why do you need to be aware of or do separation of proteins or purify proteins? So some of you have mentioned to know about structure and function of a protein. That is correct. So for example, I think Karthik has mentioned erythropoietin. So erythropoietin is one protein which is used. Insulin is another very important protein. So these are proteins which are used as recombinant proteins, and they are life-saving drugs in diabetes and in chronic kidney disease. Yeah, so you can't inject them into people unless you know about their structure and function, right? So you need to manufacture recombinant proteins, and unless you know the details about their structure and function and their conformation and their other details, you will not be able to raise recombinant proteins. One. Second is, as some of you mentioned, diagnosis of disease, but now as we are moving deeper into proteins, diagnosis of disease is a vague answer. I would like a more specific answer, so I'll just share that with you. You are right, diagnosing disease. So some abnormal proteins might be present and some normal proteins might be present in excess. So diagnostic tests or biomarkers can be developed only after you know greater details about their structure and function, right? So unless you know, suppose for example, you know that a particular protein increases in a particular disease. So to evaluate that protein, quantitatively evaluate that protein, you need to do an ELISA or some other test like that, which can actually tell you about the increased or decreased quantity of that abnormal protein, or that protein which increases and tells you that this is the disease. So unless you know about the specific structure and function of that protein, you will not be able to develop that test. Yes. Okay, is that clear? And obviously raising antibodies, developing vaccines. So all these will be possible only if you purify proteins and after you separate them from a heterogeneous mixture. Okay, is that clear? So the learning objectives of this class are that you will need to know why you are separating proteins and the basis of the properties on which different protein separation techniques are based. So we'll be talking about a few techniques and enumerating the techniques. So you should be able to enumerate the techniques by which protein separation is possible, and explain the principle of a few important methods used to separate proteins. Nobody will give you a big detailed question about protein separation, but you should know the commonly used methods used to separate proteins. For me as a teacher, I would be happy if you can answer the question why you are separating proteins, what we just discussed a few minutes back. That is more important, because you will know that the first and foremost thing when you should start a topic is why you are reading this topic. So that should be very clear to you when you're starting off, especially on a technical topic like separation of proteins or purification of proteins. Okay, so as doctors, you should be very clear why this is important for you to know and why this is important for you to study. Okay, and identifying salient features and advantages of commonly used methods. So why you use a particular method and not some other method to purify a protein. So, and finally compare and contrast between different techniques used for protein separation. Yeah. So most of you will probably be aware that recombinant insulin is used in diabetes. That probably every one of you will know. But another very interesting protein which is used is erythropoietin. So some of you will be seeing patients with chronic kidney disease, and one of the features which the kidney is responsible for is that it produces erythropoietin. So when the kidneys fail, then you have to give recombinant erythropoietin protein, inject it into the patient apart from doing a dialysis. So you have to supplement erythropoietin, and that is made possible only when you inject recombinant proteins. So these are some of the things which have come up over the years, and that is why it is very important for you to understand and know about protein separation and protein purification. Okay. Okay. So continuing from what we started off in the first two classes, we learned that the proteins or peptides have different physical properties and they're functionally different. So what scientists have done is that they take advantage of these properties and they use these properties for identifying, detecting the presence or absence of certain proteins, raising antibodies, developing analytical tests, etc. So that is why knowing about the physical and functional properties is important, and these can be used for separation of proteins as well as purification of proteins. So as we go along, you will understand how. So proteins differ in size, shape, charge, relative solubility, and obviously that also depends on pH, polarity, salt concentration, etc., as well as binding properties of proteins. So these properties of proteins, differences in size, shape, charge, relative solubility, binding properties, these are the properties which are used to separate proteins, and as we go along you will understand how. So I think Dr. Astha talked about differential centrifugation. So centrifugation is one of the very simple techniques which is used for separation of proteins. So differential means that at different speeds you will get different components coming into the product. So when you centrifuge it at different speeds, you will get different components separating out. So first I would like to just make one thing clear, that when we are talking about separating proteins, the first thing which we need is a cell lysate or a homogeneate or a heterogeneous mixture of proteins. So whether it is if you are producing those proteins in a artificial cell culture or something, so you will have to break open the cells to have a cell lysate or a heterogeneous mixture from which you will start your purification procedures. Okay? So if you have any doubts at any point of time, please put it up on the chat box, because if I don't hear from you, I'll continue and I'll assume that you are understanding what I'm saying. So the purification techniques comprise of differential centrifugation, salting out of proteins. Dr. Asta talked about salting in and salting out of proteins in details in her class on Monday using ammonium sulfate, and I'll just be briefly touching upon it today. And chromatography and electrophoresis are two commonly used techniques which you probably would have studied for your entrance exams, but we'll go into a little more detail with chromatography and electrophoresis, and some of you might have doubts. So these are concepts which need to be very clear, not mugged up, but if you understand the concept, then you can read any book and understand what they are saying. If you don't understand, it will be very difficult for you to read and understand from a book. Okay? So please ask questions if you don't understand. Now, just recapitulating what Dr. Asta said. Respirating, so I'm essentially focusing on salting out here in this class. But there is also the concept of salting in as well as salting out. So when you add salts, specifically ammonium sulfate, which has a high tendency to ionize, the salt, as the salt concentration increases, the salt ions bind to the surface charge on protein molecules, breaking open the intermolecular salt bonds between the protein molecules. So when you start initially, you'll see that proteins aggregate together by virtue of their intermolecular salt bonds, and when the salt concentration increases slowly, these salt ions will bind to the surface charges on the protein molecules, thereby disrupting these aggregates, and they dissociate and move into solution. So that is what is known as salting in. Okay? Now for today's class, I'm more interested in salting out. So when you keep on increasing the salt concentration further, Salt bonds, basically these are again type of ionic bonds which are there on the surfaces of proteins. So you have ionic interactions which are there on the surface of proteins, which constitute the intermolecular reactions between different protein molecules. So when you
Excuse me, sir.
You are not audible.
Excuse me, sir. So you are not audible to us.
वो तो मैंने म्यूट कर दिया, ये सब तो माइक ऑफ है।
उनका दूसरा वो है, उसका मुझे पता नहीं क्या है।
मतलब इन लोगों को तो साइलेंट कर दिया,
सुदीप सर को तुमने म्यूट कर दिया?
Yeah, okay. I think that was a technical problem. My ignorance. Can you hear me now? Yes. Okay.
इधर से म्यूट कर लो ना। स्पीकर ऑफ कर दिया था।
नहीं स्पीकर तो ऑफ कर दिया, अरे वो तो मैंने उस टाइम कर दिया।
So somebody had very delightedly put in chutti. So sorry to disappoint. No chutti. Back to sorting out. Okay.
तुमने सर को म्यूट किया था लग रहा है।
So yeah, salt bonds are essentially bonds which are ionic interactions, which are not only ionic interactions but other interactions as well. But yes, obviously there are ionic interactions and other non-covalent interactions which bind proteins. Yeah, so there are.
यूट्यूब का इशू नहीं है, ये बंद हो जाएगा। यूट्यूब वाले का मनोहर जी बोल रहे हैं कि उसको बंद नहीं कर सकते। ये क्या चीज़ है? ये क्या है? क्वेश्चन में
मुझे पता नहीं ये कैसे है। टेबल्स लेके आता हूँ।
ठीक है।
39 होंगे ना यहाँ पे, सॉरी 41 है टोटल। 41 बच्चे हैं टोटल, ये देखो ये ठीक है। 56 to 96 41 हुआ है ना, ठीक है। तो हर तो 41 है, इसमें भी 41 है। 41
ऑडियो ऑप्शन
कुछ नहीं कर सकता
हमारे क्लासेस चाहती है सब पढ़ा दिया जा रहा है और हम लोग आ जा रहे हैं तो ऐसे जा रहे हैं। अरे आप 2 मिनट पहले फिर बोला जाएगा तुम वीडियो के साथ क्यों नहीं पढ़े, इन लोगों को क्यों काम कर रहे हैं, समझ आ रही है।
मुझे पता नहीं।
और ये बैग्स लेके अंदर क्यों आ रहे
Keep your bags here. Please be silent. There is a first year MBBS lecture going on here through board meeting. Okay, we don't know what is going there, so please. So we don't have permission to use this lecture right now, but we are using it. So please remain silent. Okay. Please remain silent. Keep your bags here and stay at your place. You can sit accordingly however you like.
Please keep silent. There is a class going on here and your voice can be heard. Please cooperate, everybody. Keep your bags open. Try to make sure you don't have any paper or mobile
first you will get 10 minutes for that. After that we will correct the MCQ answer. Okay? So make sure you finish MCQ first. After that you will have your SAT. Keep your own answer. If you find anyone cheating, you will bring the answer. Okay. Not only for the MCQ, but also for the MCQ.
Any student of batch A or C sitting here? All of you are from batch B? Okay, ma'am. Please don't forget to write your roll numbers clearly.
not half an hour. Total time is 30 minutes. For MCQ we have 10 minutes and SCQ is 20 minutes. We have 3 more minutes for MCQ. Your roll number or MCQ paper. You're okay. Time is up for MCQs. You will return your MCQ answer sheet. Anyone who hasn't returned MCQ answer sheet? Has anyone not returned MCQ answer sheet? You will not be collecting after this, okay? MCQ. से एक काम करना, जो एक पेपर में है ना, ये लिखना है कि डेमो रूम इज बुक फ्रॉम 3:30 to 5 ऑन, आज का डेट डाल दो 17/12। डेमो रूम इज बुक फ्रॉम 3:30 PM to फ्रॉम 3:30 हाँ 3:30 PM to 5 PM on 17/12/2020। ठीक है, वो डेमो रूम के बाहर चिपकाना है। ठीक है, ऐसे ही एक वाइट पेपर में लिख के चिपका दो।
Roll number 64.
कोई प्रॉब्लम होगा मॉर्निंग सेशन
अभी ठीक है।
नहीं हमारा तो सही चल रहा है, शुरू से ही चल रहा है, सुबह से ठीक चल रहा है।
अभी ठीक है।
हो सकता है अभी हफ्ते भर से तो आया मैं दोस्तों नया नया तो
इधर ही जब लेकिन फिर वो एक जगह ट्राई करने में हो गया।
मेरा नंबर है आपके पास?
आपका नाम क्या है?
सुशील
सुशील। मेरा नाम है विंसेंट। मैं ऐसा
भाई का इस्तेमाल करूँगा विल्सन
विंसेंट
विल्सन
विंसेंट VINCENT।
विन्नी
सुशील है ना?
मैं किसी को सेल कर देता हूँ डॉक्टर वगैरह डॉक्टर के नाम से नहीं होगा मतलब इसके नाम से आ नहीं रहा इसमें वो बोलेंगे
वो पता नहीं।
ठीक है नाम है? अल्पना। क्या है? हो गया? अल्पना शर्मा सबसे ऊपर देख लीजिए।
मैम को ना वो प्रेजेंटर बना दो, मैम वहाँ रूम के सामने बैठी हुई है।
हाँ वो नाम का अल्पना शर्मा होगा इधर। एक मिनट दे दो। अल्पना मैम का नाम क्या है? मैंने ज्वाइन नहीं करा ना, ज्वाइन करवा दो।
एक बार आके ज्वाइन करा।
मैं एक बार कर लिया मैम ने ज्वाइन
कर लिया बट
यहाँ पर शो ही नहीं कर रहा। एडमिनिस्ट्रेटर आ रहा है।
हाँ
नीचे
वो ना वो लिखा है वही है।
हाँ
वो तो आप तो कह रहे थे अल्पना के नाम से
मुझे भी
नीचे ही है वो।
हाँ नीचे जाओ। नहीं आप अभी भी नीचे
आ रहा था स्टार्टिंग में आ रहा था वो तो
सबसे लास्ट में।
हाँ चलो नीचे वो तो मैं कभी कर देता
ये
हूँ।
उनको कर लो। हाँ इसमें क्लिक करो। नहीं प्रेजेंट है। यस ठीक है। ये
वो तो मैम को बना कर दिया।
हाँ हो गया। लिख रहे एक मिनट। हेलो हाँ एक बार
एक मिनट मैं कर रहा हूँ।
मैम प्रेशन करते हुए। हाँ हाँ मैम को स्टार्ट करने के लिए बोल दो। स्लाइड से दिख रहा है।
टाइम तो 3:30 मिनट्स है, 30 मिनट्स लगेगी। ठीक है।
क्या करना है?
हो गया।
ओके ठीक है।
मैसेज आ रहा है।
हाँ वो तो
हेलो
So, I am Dr. Alpana Sharma and this class is on plasma proteins and their functions. उसका do not disturb लगा देना. So, you must be knowing what is plasma and what are proteins. We are studying proteins for last 2 days, separation yesterday and other topics. So, not able to move my slide. Just a minute, I'm not able to move my slides. Yes, now I am. Santosh ko bhejo. So the objectives for today's class are to know the blood components. Slide ये screen को side नहीं हो रहा है. So the objectives of today's class is, after this class you should be able to know the blood components, you should be able to know the different plasma proteins and to understand the various functions of these plasma proteins. And finally you should be able to know the associated diseases with low or high levels of these proteins. So first of all, we should be able to differentiate between what is the term blood, what is the term plasma, and what is the term serum. In the practical classes you will be taught in detail, but here I would like to tell you that whole blood, which is a body fluid, we can collect it by arterial or venipuncture with anticoagulants like heparin, EDTA or sodium fluoride, depending what investigation you want to perform. So whole blood means we are obtaining the blood in the anticoagulant vial. What is plasma then? The plasma is when the whole blood which is being collected in anticoagulant is centrifuged at 2000 rpm, then the supernatant or the fluid on the top is plasma. So centrifugation, that also you will be seeing in your practical class. Every day you will use centrifuge. So the centrifuge is a machine where we can spin down the solution, where cells will settle down, and the fluid, the supernatant, is plasma, which will be on the top. Then what is the difference between plasma and serum? Serum, when we collect serum, it will be collected from the blood which is being collected in a plain container. We won't use anticoagulant. So the blood collected in plain glass container without any anticoagulant, and then we centrifuge after some time at 2000 rpm, and then the supernatant is serum. So plasma is being separated from blood which has been collected with anticoagulant, and the serum is the fluid which has been collected after centrifugation from the blood which has been collected in plain vial, no anticoagulant. So these are the differences between blood, plasma and serum. So today we'll be learning about plasma protein. So what are the functions of blood? Everybody knows it that blood has many functions, and the functions of blood are mainly carried out by plasma and its constituents. Plasma and its constituents means plasma has many constituents, but today we will be focusing on plasma protein. So except for some specific cellular functions like oxygen transport, cell-mediated immunologic defense, the major function of blood is carried out by plasma and its constituents. Plasma consists of water, electrolytes, metabolites, nutrients, proteins and various hormones. So it has many constituents. Today we will be focusing on proteins, and in their subsequent classes, day by day, when you will learn metabolism, you will come to know what are different metabolites, what are different hormones, where are they present. And the water and electrolyte composition of plasma is almost same as that of extracellular fluid. So it is almost same as we have in the all extracellular fluids. And you also must be knowing that laboratory determination levels of these small these ions like sodium, potassium, calcium, chloride, bicarbonate, they are very important in many of the management of diseases. So what are the major functions of blood? It has many functions, as I told you. Respiration an important function, where transport of oxygen from lungs to the tissues and of carbon dioxide from the tissues to the lungs. So that is respiration, that is the transport of oxygen. Then nutrition, transport of absorbed food materials. This is also being done by blood. Excretion, transport of metabolic waste to the kidneys, lungs, skin and intestines for removal or for excretion. Maintenance of normal acid-base balance in the body. The acid-base balance in the body is mainly maintained by the lungs, by H+ and bicarbonate ions. This will also be taught in subsequent classes. So it also maintains the normal acid-base balance, otherwise there will be acidosis or alkalosis, that also a full class. Regulation of water balance, which is through the effects of blood on the exchange of water between the circulating fluid and the tissue fluids. Regulation of body temperature is also an important function of the blood by distribution of body heat in our body. Then defense against infection by white blood cells and circulating antibodies. This is also an important function performed by the blood, where there is a defense against infection by WBCs or by circulating antibodies. Transport of hormones and regulation of metabolism, transport of metabolites and coagulation. So there are various functions of the blood which are listed here, but we'll be focusing mainly on the plasma. So what is plasma? Plasma contains a complex mixture of proteins. The total protein concentration in human plasma is between 7 to 7.5 grams per deciliter. It is not milligram, this is grams per deciliter. The proteins of plasma are a complex mixture of simple and conjugated. Simple means individual proteins, conjugated means, as you will see the name, glycoproteins, and types of lipoproteins. Glycoproteins mean some carbohydrate moiety and the protein. Lipoproteins means lipids and proteins. So in the plasma we have simple proteins as well as conjugated. Thousands of antibodies are present in human plasma. The relative dimension and molecular masses of most important plasma proteins are different. Some are small, some are big. The separation of individual plasma proteins, as just now you have learned the separation of proteins, can be done by different methods. One can use the solvents or electrolytes, or by solvent fractionation we can remove different protein fraction on the basis of their solubility characteristics. That can be salting out method, which is important in the determination of different protein fraction in the clinical lab. And thus one can separate the proteins of plasma by the use of varying concentration of salts, that is sodium or ammonium sulfate. So here you can see, these pictures are given in Harper. You can see the size as well as the dimensions of different proteins present in the plasma. Albumin, we can call it 69,000 Dalton or 69 kilodalton. So the molecular weight of albumin is 69 kilodalton. Hemoglobin is approximately 65 kilodalton. Beta 1 globulin is 90 kilodalton. Gamma globulins are 156 kilodalton. Alpha 1 lipoprotein is 200 kilodalton. Beta 1 is 1300 and fibrinogen is 350 kilodalton. The biggest, the highest molecular weight in the plasma protein is of fibrinogen. So albumin is the least, which is 69 kilodalton, and which is an important component of plasma. So the most common method of analyzing plasma proteins is by electrophoresis also. There are many types of electrophoresis, each using a different supporting. What is electrophoresis? As this term says, the separation of molecules on the basis of their charges when we pass the electric current. And this is being done on a different supporting medium, where we can use gel, we can use paper. So in the clinical laboratory, cellulose acetate is widely used as a supporting medium. Cellulose acetate is nothing but it is a filtered, refined filter sheet, we call it Portman filter sheet. You must have seen in your 12th class laboratories, practical classes, the filter sheet. So the refined filter sheet cellulose acetate paper. Generally in the clinical lab they use cellulose acetate paper as a supporting medium for electrophoresis. So after doing the electrophoresis on cellulose acetate membrane, after staining, these plasma proteins dissolve into 5 bands: albumin, alpha-1 fraction, alpha-2 fraction, beta and gamma fraction respectively. These stained paper can also, these stained strips of cellulose acetate is called an electrophoretogram, and the amounts of these bands after separation by electrophoresis on this cellulose acetate, the amount of these bands can be quantified. And we have different machines like densitometry scanning machines or densitometer, where we can find out the amount of these bands or the concentration of that particular fraction in the sample. So the characteristic changes in the amounts of one or more of these 5 bands are also found in many diseases where alpha-1 fraction is high or albumin is low. They are many times associated with diseases. So this is just the picture, that this is the cellulose acetate paper where we can load our sample here, and then we can dip this paper in the buffer, the transfer buffer, and then connect with these electrodes, positive and negative, and then we pass the current. So after that, there is a separation into albumin, alpha-1, alpha-2, beta and gamma fraction, and after densitometry we can find out the concentration of the particular fraction. We know in a normal situation albumin is the 60% of albumin is present in the plasma. So it has major fraction. So proteins with a negative charge will migrate toward the positively charged anode, and albumin has the most negative charge and will migrate furthest towards the anode. So that's why this is the first fraction, that is the albumin. Then alpha-1, alpha-2, beta and gamma. And they were named accordingly, like which fraction is coming first. So how these plasma proteins are synthesized? As the protein synthesis takes place on the ribosome, plasma proteins extensively studied in both humans and animals. Their biosynthesis, structure, function, all are almost known. They are synthesized in the liver on membrane-bound polyribosomes. They then traverse the major secretory route in the cell, like from RER to smooth endoplasmic membrane. From there they go to Golgi apparatus, and then the secretory vesicles, then they enter into the plasma. So this is the route, like traverse through the major secretory route from RER to SCR, and then from the Golgi apparatus and finally into the secretory vesicle before entering into the circulation. Most plasma proteins are synthesized as pre-proteins and initially contain amino-terminal signal peptide. And we know that once these proteins are synthesized, after that there are some modifications which we call post-translational modifications. That also you will learn. So like proteolysis, we trim the protein, glycosylation, we add some carbohydrate moieties, or phosphorylation, like for the proteins are phosphorylated. So these are all post-translational modification. So they occur as they travel through the cell. The transmit time through the hepatocyte, because they are synthesized in the liver, from the site of synthesis to the plasma vary from 30 minutes to several hours for individual proteins. Some proteins they take less time, synthesize and come in the plasma. Some proteins take more time. Most plasma proteins are glycoproteins, meaning they are glycosylated, either N-linked or O-linked oligosaccharide chains or both. N-linked means the amino acid which have amino, as there are O-linked which have OH, like tyrosine. So some of the amino acid, they asparagine as for N-linked glycosylation, tyrosine for O-linked glycosylation, where some amino acid, designated amino acids, they are linked with the oligosaccharide chains, and hence plasma proteins becomes glycoprotein because they are glycosylated. But albumin is the major exception. Although it has the largest amount in the plasma, but it does not contain sugar residues. This is non-glycosylated. So here you can see just this diagram tells what are the concentration of respective fractions of different plasma protein. So albumin has 60% contribution. Then the IgG, we have transferrin protein, fibrinogen, alpha-2 macroglobulin, alpha-1 antitrypsin, haptoglobin, alpha-1 acid glycoprotein, and different apolipoprotein, the conjugated proteins and others, and some other medium and lower witness proteins, they also contribute in the plasma. So the major 60% is albumin, then the immunoglobulins, then other proteins which have different fractions. So this is also one important thing, that plasma proteins, not all but mainly, mostly, they are polymorphic in nature. They exhibit polymorphism like alpha-1 antitrypsin, haptoglobin, it means they have some allelic variation, transferrin and immunoglobulins. They exhibit polymorphism, and the polymorphic forms can be distinguished by electrophoresis or isoelectrofocusing. You know what is isoelectrofocusing? Where the separation is based on this PI, this isoelectric point. Each form show a characteristic migration. So then we come to know what is the half-life in circulation of this major plasma protein. It can be determined by labeling the protein with I-131, that radio label. And this isotope unites covalently with the tyrosine residue in the protein, and then one can find out what is the half-life of that particular plasma protein. After some time you can see, so that if they are degrading, then their radioactivity will be lower and lower. So the half-lives in normal healthy adult is 20 days for albumin and 5 days for haptoglobin. It means that all plasma proteins are not having the same half-life. They have different half-life, like albumin 20 days, but haptoglobin is just 5 days. In these proteins, the plasma proteins, some of the groups are specific for some of the protein, like in ceruloplasmin, we have some sialic acid residues also. So if we remove the terminal sialic acid, then and we expose it to neuraminidase, it can shorten their lives in the plasma. So half-life can be shortened, sometime maybe 1 day or 2 days. And some gastrointestinal diseases such as regional ileitis, amount of plasma protein decreases in these diseases. And then the patient have some condition like protein losing gastroenteropathy, where half-life may be reduced as little as 1 day. So this is the list which is given in Harvard, where different functions are given and the plasma proteins are there. Today we will be discussing the important plasma protein functional point of view and for you. So antiproteases, we will be discussing alpha-1 antitrypsin and alpha-2 macroglobulin. Then blood clotting proteins are there, then enzymes, hormones, this we will not discuss. Immune defense, yes, immunoglobulins are there, beta-2 microglobulin, and then transport or binding proteins like albumin, ceruloplasmin, haptoglobin, and transferrin will be discussed. So there are many proteins which are present there, but we will be discussing some. So albumin, the important one, when we ask about question given for what are plasma proteins, we have to first mention albumin. The albumin is 69 kilodalton and it is the major protein of human plasma. So 7 to 7.5 gram per deciliter total protein, and here the concentration of albumin in the human plasma is 3.4 to 4.7 gram per deciliter. It means it makes up approximately 60% of the total plasma. The gene for albumin, which codes for it, is located on chromosome number 4, and about 40% of albumin is present in plasma, and the other 60% is present in the extracellular space. Liver produces about 12 grams of albumin per day, which represent about 25% of total hepatic protein synthesis. It means the liver produces 25% this albumin, which is the 25% of total hepatic protein synthesis, and half is secreted protein. Albumin is initially synthesized as a pre-pro protein. As I told you, post-translational modification, it means when the protein is synthesized, it is in the form of pre-pro protein having a signal peptide. So if signal peptide is removed as it passes through the rough endoplasmic reticulum, and finally a hexapeptide is also removed from the amino terminal once they reach the secretory pathway. So from RER to SCR, then to Golgi, and from Golgi to secretory pathway, there again last hexapeptide is released from the amino terminal, and then this comes in the circulation, the final protein or the albumin comes into the circulation. The synthesis of albumin is depressed in a variety of disease-like levels, reduced, particularly those of the liver diseases, where there is a decreased albumin to globular ratio, the albumin is decreased. And the synthesis of albumin decreases relatively early in the condition of protein malnutrition, such as kwashiorkor. There also the synthesis of albumin decreases. So, what is important here? That albumin, which is 69 kilodalton, is a major plasma protein, approximately 3.4 to 4.7 grams per deciliter concentration, and this liver produces 25% of total hepatic protein, which is albumin, and it is initially synthesized as pre-pro protein. First, the single peptide is removed once it passes through the rough endoplasmic reticulum, and later on a hexapeptide is cleaved off from the amino terminal along the once it reaches from the secretory pathway, and then it comes as a final albumin protein in the circulation. So mature human albumin consists of one peptide chain of 585 amino acids, single polypeptide chain of 585 amino acids, and it contains 17 disulfide groups. So rich in cysteine residue form disulfide. By use of proteases, scientists have studied that albumin can be subdivided into 3 domains which have different fractions. So you know, if we have a protein polypeptide of 585 amino acid, it is never present in a straight form. It will acquire its conformation at the 3-dimensional structure. So by the proteases, one can find out that yes, the albumin has 3 functional domains, or the loops, or the important conformation, which have different functions, these domains. The 3 homologous domains, almost same, assemble to form a heart-shaped protein. So 2 domains on top and 1 on the later here. So each domain is a product of 2 subdomains, A and B, that possess common structural motifs. And the principal region of ligand binding, because albumin function as a transporter or the transport various ligand binding to albumin are located in the hydrophobic cavities in the subdomain 2A and 3. So 2A and 3 means domain 2A subdomain, domain 3 subdomain A. So this is actually the ligand binding site which has a hydrophobic cavity in the subdomains 2A and 3. Structurally, the serum albumins are similar, each domain containing 5 or 6 internal disulfide bonds. So since we have 17 disulfide bonds, it means each domain have 5 to 6 disulfide bonds. It has an ellipsoidal shape, it is not straight, it is not elongated, hence does not increase the viscosity of plasma. as an elongated molecule fibrinogen does. Fibrinogen is an elongated structure as we have seen in the initial slide, but the albumin is an ellipsoidal shape and does not increase the viscosity of the plasma. So as we were discussing, it has three domains, 1, 2 and 3, yes?
Ma'am, if albumin is ellipsoidal in shape, then what is the heart shape formed by the three homologous domains?
This is the conformation, three-dimensional structure. Finally, the albumin. This is a heart shape. This is the conformation which we have found from the X-ray crystallography. This structure, but when we see, this is in the ellipsoidal shape. So one can form the ellipsoidal shape like this also. So this is a three-dimensional shape where the three-dimensional structure of albumin, where we have three domains, 1, 2 and 3. Each domain have subdomain 1 and 2, A and B, like same here and like same here. N terminus here, C terminus. This is extra crystallographic structure. The detailed structure of the functional domains present. So once we bind the different drugs or any ligand, this is the cysteine residue SH portion which actually binds with different drugs. That's the example that how this drug can be bound to this albumin molecule. So cysteine 34th position of circulating albumin and accumulates in the solid tumor. So this is a drug which can bound to SH or the cysteine residue of this. So since albumin has relatively low molecular mass, just the 69 kilodalton, and it has high concentration as 3.4 to 5.4 or whatever it is, albumin is responsible for 75 to 80% of osmotic closure of human plasma. The important function of plasma protein albumin, which maintains the osmotic or oncotic pressure, or we can call colloid osmotic pressure, which is a form of osmotic pressure which is exerted by proteins, mainly the albumin, in a blood vessel's plasma that usually tends to pull water into the circulatory system. So this albumin, low molecular mass, high concentration, it maintains the oncotic pressure, and where it is being exerted by albumin in a blood vessel's plasma that usually tends to pull water into the circulatory system. And this colloid osmotic pressure is just the opposing force to hydrostatic pressure, because this hydrostatic pressure, it increases filtration by pushing fluid and solute out of the capillaries where it is pulling into the circulatory system. So this albumin maintains the oncotic pressure in the blood vessels. So the other functions are transport. It acts as a transporter or transport function. Its ability to bind various ligands and act as transporter. These include free fatty acids, calcium, certain steroid hormones, bilirubin, etc. So it can transport many molecules or various ligands. In addition, it plays an important role in transport of copper also in the human body. So it can transport many molecules. A variety of drugs, including sulfonamides, penicillin G, aspirin, they are bound to albumin as important pharmacological implications. If you are taking the drug, it will bind to, this will be transported through albumin. So and people have tried this in hemorrhagic shock or in burns cases. They have given human albumin as an experimental for the treatment, but later on they have found that there are some recent studies which have suggested that administration of albumin in these conditions may increase mortality rate. So, sorry. So this is in the experimental phase, but yeah, people are trying. Sorry, just one. This is a little dry topic, but yes, important. Next comes the haptoglobin. What is haptoglobin? It is again a plasma glycoprotein. As I told you, albumin is non-glycosylated protein. The others are glycosylated protein. So plasma glycoprotein means it is glycosylated and binds the extracorpuscular hemoglobin in a tight non-covalent complex, like it forms HbHb, hemoglobin and haptoglobin, non-covalent complex. It prevents free hemoglobin from entering the kidney for excretion. It means haptoglobin concentration in human plasma ranges from 40 to 180 mg per deciliter. We know that approximately 10% of the hemoglobin degraded each day is released into the circulation, and thus it is called extracorpuscular hemoglobin. So the other 90% is present in the old damaged RBCs, which are degraded by cells of the histiocytic system, that is the reticuloendothelial system. But the 10% of the hemoglobin which is degraded each day released into the circulation, and which forms the non-covalent complex with the haptoglobin. The molecular mass of hemoglobin is approximately 65 kilodalton as we discussed, whereas simplest polymorphic form of haptoglobin, that is HB11, we will find out that yes, just see that it has three polymorphic form, which is found in human is approximately 90 kilodalton. So haptoglobin is 90, hemoglobin 65. So it forms a complex of a molecular mass of approximately 155 kilodalton. And this complex is little too large to pass through the glomerulus. It means it keeps the hemoglobin in a complex form which has not been excreted. So this haptoglobin conserves iron present in the hemoglobin, otherwise it has been lost. It consists of 2 alpha and 2 beta chains, in which are linked by disulfide. The haptoglobin have 3 polymorphic forms: HB11, HB21 and HB22, because haptoglobin has 2 genes which are designated HB1 and HB2. So they direct these 3 phenotypes. If HB11 is there, it will be homozygous phenotype. If HB22, again it is a homozygous phenotype, so it has just 2, and but HB21 is a heterozygous phenotype. The levels of haptoglobin in human plasma vary and of diagnostic use, because how much hemoglobin it is taking care of. So the low levels of haptoglobin are found in patients of hemolytic anemia. What does it mean? In hemolytic anemia, more and more this hemoglobin is coming out, so more and more HP is forming the complex form. The free haptoglobin will be less. Half-life of haptoglobin is 5 days, but once it forms a complex with the hemoglobin like HbHb, this complex takes 90 minutes. Just 90 minutes for the degradation. So complex being rapidly removed from the plasma by hepatocytes, thus bound HbHb is cleared from plasma 80 times faster than the normal haptoglobin. So levels of haptoglobin falls rapidly, where hemoglobin is constantly being released from the RBC, as occurs in the hemolytic anemia. It is an aqueous phase protein and its plasma level is elevated in a variety of other inflammatory states. So here you can see just the representation of what we have learned. The haptoglobin synthesized in liver, it has 2 alpha chain, 2 beta chain linked with disulfide form. So what is happening in the cell when the RBCs are hemolyzed? Then hemoglobin is released, which forms Hb dimers also. And then once we have haptoglobin here, then this haptoglobin forms complex with this hemoglobin dimers. And once this complex is formed, then on the macrophage's surface we have one CD163 receptor, which recognize this hemoglobin haptoglobin complex. complex is endocytosed, this complex inside, and then this complex is.
Excuse me, ma'am.
Yes. Yes.
And what is the meaning of half-life of a protein?
Half-life of protein is degraded after some time. It cannot live for long, for like defined period. No, indefinite period. It cannot. It has to be degraded after some time. Okay. So every protein has its half-life. Yes.
You're fine, ma'am. Thank you.
Okay. So once the hemoglobin complex is formed, it is being recognized by the CD163 receptors which are present on the macrophages, and this complex is endocytosed, and then it forms the endolysosome where the pH decreases because we have mainly the acid enzyme, the acidic environment in the lysosome, and then it is being degraded. So and these at low pH, this complex is released from the receptor, and these receptors are recycled back. So this way the haptoglobin takes care of conserving iron, utilizing iron, which is being in the circulation by the RBC hemolysis, approximately 10% from the complex taken up by the cells through the endocytosis, through this receptor mediated endocytosis, and then it is degraded at units. The third protein, so we have albumin and then haptoglobin. The transferrin, as the name suggests, transferrin, it is a beta globulin with a molecular mass of approximately 76 kilodalton. It's a glycoprotein again and synthesized in the liver. About 20 polymorphic forms of transferrin have been found. It plays central role in transporting iron around the body. So it transferrin transports iron in the body. It transports iron, the two molecules of ferric ion, Fe+3 per molecule of transferrin. So one molecule of transferrin can transport two molecules of this iron in the ferric cell. In the circulation to sites where iron is required, from the gut to bone marrow and to the other organs. So this transferrin can transport iron to various places. It is important because its occurrence in many heme proteins is there. They're like hemoglobin, it requires iron, myoglobin requires iron, and cytochromes. So that also you will learn in the due course. Once your classes will be on, all the topics will be covered. Anyway, hemoglobin I will be covering in next classes. So it is ingested in the diet either as heme or the non-heme iron. So this is just the picture of transferrin, how it takes the iron or Fe+3. So what happens? We have transferrin receptor on the cell surface, and these receptors are clathrin present on the clathrin coated pit. What is clathrin coated pit? Clathrin coated pits are nothing but the pits, they have the clathrin protein on the surface of these pits. And once the receptor ligand complex is formed, then it forms a complex. Clathrin is released, and then the endosome is formed. And finally, there is a release of hormone. So what is happening here? We are having Fe+2, which transferrin has taken it, and then it binds with the TFR receptor or the transferrin receptor, and then these transferrin these transferrin receptor, they form a complex having the Fe+3 two molecules in one transferrin molecule, and then once it is formed, the endosome taken up by the cells, and then by the acidic environment, what happens? In the acidic environment, these iron, the acidification releases this Fe+3, and later on they subsequently reduce to Fe+2. It is little different than LDL receptor that you will learn low density lipoproteins, which is also being taken up by the clathrin mediated, clathrin coated pits, the endocytosis, but there the LDL is broken down. But here the transferrin is still holding by the receptor, only the iron is released at low pH, and this later on subsequently reduced to Fe+. And this complex which is goes back to the surface and the recycled back, and once they reaches on the surface, then only the transferrin is released from the receptor. So this is little different. Here the receptor and transferrin, they form the complex and then they reaches to the surface after the recycling. So transferrin shuttles iron to various sites of the body, and here the approximately 200 million RBCs, or about 20 ml are catabolized every day, releasing around 25 milligram of iron into the body, most of which will be transported by transferrin. So transferrin has important function, and there are receptors. The picture which we have seen is just written in the text form. Just to revise, that there are receptors on the surface of many cells of transferrin. It binds to these receptors and internalized by a receptor mediated endocytosis. Acid pH inside lysosome causes the iron to dissociate from the protein. The dissociated iron leaves the endosome via DMT1 or divalent metal transporter 1 to enter into the cytoplasm. Unlike protein component of LDL, FODF is not degraded in the lysosome. Instead, it remains associated with its receptors, turns to the plasma membrane, dissociates from its receptor, and re-enters into the plasma, picks up more and more iron, and again delivers to the iron release. So this is recycling back, and again it is ready to take 2 molecules of Fe+3 again, and then transfer it to wherever it is required. So along with transferrin, transferrin is transferring the iron to different places, whereas ferritin stores iron in the cells. So here the ferritin is another protein that is important in the metabolism of iron. It is the concentration is less in human plasma. However, in patient with excess iron, the amount of ferritin in plasma can be elevated. Under normal conditions, it stores iron, ferritin. In conditions of excess iron like hematochromatis, body stores of iron are greatly increased, and much more ferritin is present in the tissues such as liver and spleen. And ferritin contains approximately 23% of iron, and apoprotein ferritin, that is protein moiety free of iron, has a molecular mass of 440 kilodalton. It is a long molecule like 24 subunits of 18.5 kilodalton each, which surround in a micellar form some 3000 to 4500 ferric atoms. So these 24 subunits, they form a micellar structure, and where the 3000 to 4500 ferritin atoms can be stored. So the amount of ferritin in plasma can be conveniently measured by a sensitive and specific radioimmunoassay or ELISA, which serve as an index of the body iron stores. The important thing here is that synthesis of transferrin receptor and that of ferritin are reciprocally linked to cellular iron content. One is transferring, another one is storing. So here their expression is regulated. They are like alternately regulated. If transferrin receptors are having more and more expression, they are synthesizing more and more transferrin receptor, it means ferritin receptors will be lowered, because if it is transferring iron to various places, means ferritin storage is not required. If storage is more, then the transfer is not required. So in contrast, when iron levels are low, the transfer receptor mRNA is stabilized. An increased synthesis of receptor occurs while ferritin mRNA is apparently stored in an active form. This is an important example of control of expression of protein at the translational level. Both are synthesized, but their expression will be regulated. Then comes the ceruloplasmic. It is about 160 kilodalton, is an alpha-2 globulin. It has blue color because of its high copper content. It transports 90% of copper present in the plasma. Each molecule of ceruloplasmin binds 6 atoms of copper very tightly so that copper is not readily exchangeable. Albumin carries just other leftover 10% of plasma copper but binds the metal less tightly than does ceruloplasmin so that it can donate copper easily to the tissues than ceruloplasmin and appears to be more important than ceruloplasmin in copper transport in the human body. That's why the albumin has important function to transport copper also that we have learned there. The amount of ceruloplasmin in plasma is decreased in liver diseases, and low levels of ceruloplasmin are found in Wilson disease, which is due to the abnormal metabolism of copper, as well as one more disease, the Menkes disease, another condition involving abnormal copper metabolism. So, abnormal copper metabolism can have two diseases, the Wilson and the Menkes disease. So, copper is a cofactor for certain enzymes. That's why it is important, essential trace element. We know that it is metal cofactor for variety of enzymes. It accepts and donates electrons and is involved in reactions involving dismutation, hydroxylation and oxygenation. It means superoxide dismutation reaction, various hydroxylation reaction, various oxygenation reaction. This all you will learn. But yes, here I have to tell that yes, copper accepts and donates electron and involved in reactions like dismutation, hydroxylation and oxidation. And if we have excess copper, then it can oxidize proteins and lipids. Oxidation of proteins and lipids can cause free radical degradation. The oxidative stress, it can bind to the nucleic acid and enhance the production of free radicals. So excess copper is not good. The body of normal adult contains about 100 mg of copper, located mostly in bone, liver, kidney and muscle. This is a dietary intake, and copper is carried to liver bound to albumin taken up by liver cells, and part of it is excreted in the body. So we have just, yes, the Menkes disease, which is due to defect, and it is due to mutation in the gene encoding a copper binding P-type ATP. This is an important enzyme which requires ATP for the energy, and it is a copper binding P-type ATP. You will learn again in different metabolic reaction. So Menkes disease, and it is reported in 1993 that yes, the mutation of this gene encoding this particular enzyme can cause Menkes disease, which is classical kinky or steely hair. It's a disorder of copper metabolism. It is X-linked. It means affects only male infants. Involves nervous system, connective tissue and vasculature, and usually fatal in infants. It is responsible for directing the efflux of copper from the cells, and when gene is mutated, like which is happening in this particular copper binding P-type ATPase gene, then copper is not mobilized normally from the intestine and it accumulates and does a variety of other cells and tissues, and from where it cannot exit. So there is an accumulation of copper in different tissues. The next one is alpha-1 antitrypsin or alpha-1 antiproteinase. The alpha-1 antiproteinase is important. 52 kilodalton. Yes.
Then why only means are affected in that disease, Menkes disease?
Because this is the important binding P-type ATPase. If mutation is there, this is the important enzyme in the metabolism when you will learn the copper metabolism. So if this enzyme is not there, then there will be accumulation of this particular copper in different tissues. So it can cause the mental retardation, connective tissue defect, vasculature, etc.
But its gene is X-linked, so females can also be affected, right?
So this is X-linked. So okay. Sorry. This is not only in infants. It can affect. Sorry, I think I have it. So antiproteinase is a single chain protein of 394 amino acids contain 3 oligosaccharide chain. I think I have a little faster. Already 4. You have next class from 4 to 5. Sorry, can anybody tell me? No, so I'll just take 4 to 5 minutes. Sorry? All are exhausted?
No ma'am, you can continue the talk.
Yeah. I'll take 5 to 10 minutes. 5 to 7 minutes, not 10. And is the major component 90% of the alpha-1 fraction of the human plasma, synthesized by hepatocytes and macrophages. Serine protease inhibitor of human plasma. It inhibits trypsin, elastase and certain other proteases by forming complexes with this particular alpha-1 antitrypsin. Alpha-1 antitrypsin means against trypsin, proteinase. So why it is important? So you can see here, like we have different enzymes, elastase or this. So if we have active elastase and alpha-1 antitrypsin is present, what will happen? It will inactive elastase. And alpha-1 AT complex will be formed and no proteolysis of lung will take place and no tissue damage. But if the active elastase have no alpha-1 antitrypsin, what will happen? Active elastase will be here. This active elastase will proteolyze the lungs and then there will be tissue damage. That's why this alpha-1 antitrypsin is important. A deficiency of this protein has a role in certain cases, like approximately 5% of M5C. This is a disease where the tissue damage takes place, mainly the lung, because there the active elastase is not being inhibited, hence it proteolyzes the lung tissues and there is a tissue damage. So this is also important protein. The next one is the large plasma glycoprotein alpha-2 macroglobulin, 720 kilodalton, 4 identical subunits of 180 kilodalton. It comprises 8 to 10% of the total plasma protein in humans and it transports zinc in the plasma. Okay, so 10% of zinc in plasma is transported by alpha-2 macroglobulin. So one iron transport, another copper transport and third is the zinc transport. So alpha-2 is a macroglobulin which transports zinc. Then the protein is synthesized by a variety of cell types including monocytes, hepatocytes and astrocytes and is the major member of group of plasma protein that includes some complement proteins also like C3 and C4 that will be taught in your complement class. And then it binds many proteinases and is thus an important pan-proteinase inhibitor. And it forms a complex and rapidly cleared from the plasma by a receptor located on many cell types. So this is important, which is for the zinc transport. Then comes the plasma immunoglobulins. That you must be knowing that these are the defense. This is the immune system of the body, which consists of 2 major components: B lymphocytes and T lymphocytes. B from bone marrow, T from thymus. And these B cells are responsible for the synthesis of circulating antibodies, the tumoral antibodies of the immunoglobulins. And these globulins are present in our plasma. And these plasma immunoglobulins are synthesized mainly in plasma cells. And these are specialized cells, B cell lineage, and that can secrete immunoglobulins and which can take care of variety of foreign antigen and can defend our body from various infections or the pathogens or the foreign invaders or the antigen. So plasma immunoglobulins are important. We know plasma immunoglobulins, we have different 5 classes of immunoglobulins. And immunoglobulin general structure have 2 heavy chains and 2 light chains. Heavy chains are 50 kilodalton and light chains are 25 kilodalton. It has amino terminal and the C terminal. The amino terminal is the antigen binding region, whereas C-terminal this performs various effector functions of the immune because it has to take care of the body from the pathogen or the foreign pathogen which binds here and forms the anti-antibody complex, and later on effector functions are mediated. It has variable region, constant region, that all you will learn in your immunology classes. Light chains have kappa chain and the lambda chain, and 5 isotypes of these heavy chains which forms 5 classes of immunoglobulin, that is alpha IgA, gamma IgG, delta IgD, epsilon IgE, and mu IgM. So it has 5 isotypes of heavy chain, and we have immunoglobulins in the circulation. This is just the diagram and the conformational structure where these domains, how they interact of the light chain and the heavy chain, that also will be taught in later classes. But you should know that yes, immunoglobulins have major fraction in the plasma. So both, whether we have overproduction of immunoglobulins or underproduction of immunoglobulins, it can result some diseases. Like we know that multiple myeloma is a neoplastic condition, and electrophoresis of serum or urine of a patient shows large increase of one particular immunoglobulin or one particular light chain. Light chain can be kappa or lambda, but most of the time it is kappa light chain which comes in the urine or present in the serum, hence termed as Benz-Jones. So if these light chains are more and more produced, monoclonal antibodies or the particular light chain kappa or lambda, if they are secreted, then this is Benz-Jones protein which is present in the 80% of the multiple myeloma. And then we have decreased production of some of the immunoglobulin classes can also cause some of the disease. Severe reduction in the synthesis of immunoglobulin class due to genetic abnormality can result in immunodeficiency disease, that is A-gamma globulinemia, where gamma globulins are not being synthesized, in which protection of IgG is markedly affected or decreased, hence impairment of body's defense against the microorganisms. And last is the complement system. This is also the part of plasma protein, and complement system is really complex. It consists about 20 plasma proteins and many fractions there it has, and is involved in cell lysis, inflammation and other process. Unlike antibodies, factors are labile when heated at 50-60 degrees centigrade, and we know that in complement, if we study that it has complement fraction 1 to 9 components, and the C9 is associated with C5 to 5-8 complex and form a membrane attack complex. This membrane attack complex involved in generating a pore in the cell membrane that causes the osmotic lysis. Hence, in the innate immunity, we will come to know when you will learn the immunology that yes, complement system plays important role, because here the cell can be lysed by osmotic lysis by making the pores on the surface of the pathogen. The inactive proteins of the system are triggered, why we call it system. The complement system is a triggered system. If it is triggered by a stimulus, then one component will trigger the next, will activate the next component. So there is a activation by proteolysis and interact in a specific sequence from 1 to 3, then to 4, 5, 6, 7, 8, 9 like that, of the other proteins of the system. This results in cell lysis and generation of peptides of fragments, and these fragments, one is of smaller fragment, one is larger fragment. So generally the smaller fragments are involved in the complement system, the larger fragments they are important for the inflammation, chemotaxis, phagocytosis and clearance of antigen antibody complex from the circulation. And you will come to know that a complement system, the activation is triggered by 3 pathways, can be triggered by 3 pathways: classic, alternative and lectin pathway. The classic pathway involves interaction of first complement component to the antigen antibody complex. Once the antigen antibody complex is formed, then only the classic pathway interaction of C1 takes place. But the other two involve direct interaction of bacterial cell surface or polysaccharides with C3b fraction, the fragment 3, where 3b fraction binds to the surface of a pathogen. So complement system have three pathways: classical complement pathway, alternative, or the lectin pathway. The classical pathway is a more recently evolved mechanism of specific active immunity. Active immunity generally we call it when the immunoglobulin side. The alternative pathway, lectin pathway, represents non-specific innate immunity. So the two pathways are in the alternate and lectin pathway represent non-specific innate immunity, whereas classic pathway, earlier it was also used to call it the innate pathway, but now it is more evolved and it is involved in the specific active immunity also. These are just the three pathways. Again, I will repeat that yes, once we learn the complement system, that time these three pathways will be taught in detail, and all three form the membrane attack complex, which makes the pores in the pathogenic cells surface, and then there is a osmolysis in the cell. They kill the pathogen. So now I would like to summarize the class. Although it's a dry class, I know एक के बाद एक proteins का नाम पढ़ते जा रहे हैं, उसका function जानते जा रहे हैं, but this is very important. Plasma proteins, albumin, transferrin, celluloplasmin, all haptoglobin, these are important. So plasma proteins. Plasma contains many proteins with a variety of functions. Most are synthesized in the liver and are glycosylated. Albumin, which is non-glycosylated, ये कई बार MCQ में आएगा क्वेश्चन. Non-glycosylated is the major protein and principal determinant of intravascular osmotic pressure. It also binds many ligands such as drugs, bilirubin, hormones, and others. Haptoglobin binds extracorpuscular hemoglobin, prevents its loss into the kidney and urine, and hence preserves its iron for reutilization. Transferrin binds iron, transported to sites where it is required. Ferritin provides an intracellular store of iron. Ceruloplasmin contains substantial amount of copper, but albumin appears to be more important for its transport. Wilson's disease and Menkes disease reflect abnormalities of copper metabolism found to be due to mutation in the genes encoding copper binding P-type ATPases. Alpha-1 antitrypsin is a major serine protease inhibitor of plasma, in particular inhibiting the elastase of neutrophils, hence cannot damage the lung tissues. Genetic deficiency of this protein is a cause of emphysema and can also lead to liver diseases. Alpha-2 macroglobulin, major plasma protein, neutralizes many proteases, targets certain cytokines to specific organs, and transports the zinc. Immunoglobulins play a key role in immune defense mechanisms of the body, and proteins of the complement system are also involved in defense mechanism against pathogens, and it has three systems: the classical, the alternative, and the lectin pathway. I think with this, the plasma protein class is over. If you have some questions, we can discuss now, and if you want to come and discuss with me, you are most welcome anytime to my room, and you can discuss it. Hello. So shall I close this lecture? Okay. If there are no questions, I am going to close it. Thank you very much. The next class will be on hemoglobin on 21st, 11 to 12. That's very important.
Thank you, God.
Okay.
Thank you.
Thank you.
Thank you.